cutest little 2027 champions out there. Well, first of all, this is my friend Chutku, and this is me, Jasmine Mam, who is your biology faculty. If at all you are someone who is probably watching this video for the first time, then a very big hi from my end, and I hope you watch this class entirely, not because I told you, but because you love the class. Yes, and also one more very important thing, that is, there is nothing in this life that is going to be impossible. So if at all you are someone who feels that I'm very bad at biology, I cannot remember things, or probably I did very bad in my 11th grade, so trust me on this, things can change if at all you put in your efforts over here, and I am pretty sure you'll do that, right? So let's go ahead with Chutku with today's chapter, that is sexual reproduction in flowering plants, and by the end of it, I guarantee you few very important things, that is best marks in your FA1, bestest marks in your second PUC boards, all concepts super clear for your KCET examination, and obviously at the end you will be the happiest, not because of just this, but because this is going to be a happy class, and I'm just assuming that this is not forceful, right? We are all friends and we are watching it because we want to watch it and we want to get good marks, right? So let's go ahead, do not skip even a beat. The times when you go like, oh my god, I'm so sleepy, you can just pause the video. I'll try my best to not make you feel sleepy. Okay, so now listen to me. When you talk about this chapter, that is sexual reproduction in flowering plants, it carries almost 13 marks in terms of your second PUC board examination. 13, okay? And in terms of KCET examination, it almost gives 3 to 4 questions every single year, which means it is important for both. What if we complete it right now and basically clear our concepts for all of it? It's easy marks for us. We do not have to struggle later on. So with that in mind, we have to start off this class, and the first part of this particular session is going to be the male reproductive system. So when I say sexual reproduction, there is male and the female gamete which are going to come together, Baba, and they're going to fuse. Fuse to form what? Fuse to form a zygote. Now we are all a zygote. Even plants also have a zygote. It's just a single cell which is made by the fusion of both of them, and they became a single cell, and this structure is known as zygote. This zygote which is there, it's gonna divide. If it was 1, it will become 2, 2 becomes 4. Now I do not have 4 hands, but 2 becomes 4, 4 becomes 8. The cells are going to increase, and later on it is going to grow into a big thing. In the case of plants, it will become a plant. In case of us, it becomes like this and this. Okay, so here when I say that sexual reproduction is happening, and when you need gametes, there should be some parts in the flowers that are going to form these gametes. In the case of a male part, okay, or basically in the case of the male gamete, who is the one which is going to produce the male gamete? The male reproductive system, which is broadly known as what? Androecium. What do you call this as? You will call this as androecium. This is a scientific name. The easy name for you guys to remember would be what? It is going to be none other than stamen. So it's just saying you stay as a men. So that is the thing, men, and that is the male reproductive part. So stamen is very important for you to remember, and the scientific name of it is what? Androecium. Cool. Now we are going to go ahead and see what is this stamen made up of. So stamen is basically made up of 2 very important things. There's one long thing like this, which is known as filament, and at the tip you have something like this, which is known as the anther. Cool. So long stalk-like structure, filament, and then this, that is the anther. You all might have heard about the pollen grains in chotu chapters. When we were kids, we used to study about pollen grains. There is a pollination that is happening over here. So the pollen grains which are there, they are not the male gametes, by the way. They have the male gametes. So these pollen grains which have the male gamete are inside the anther, and these pollen grains have to fly to their female parts, which is why usually the filament is very long, such that the anther is right over here, and when the wind comes, it carries away all the pollen grains in the anther. Good, right? Yes. So that is the basic logic out there. Now in your NCERT, few terms are written which you have to remember, and basically it goes like this. I'll tell you. So the flower which is there, you might have seen it rest on something like this, and then you have the cute little petals over here, and basically here sometimes the so these are the petals over here, right? Now this portion where the filament is attaching to the lower thing, what do you call this as, by the way? You call this as thalamus or receptacle. You call this as thalamus, or sometimes you also call this as receptacle. There is only a little difference. Sometimes we call it same thing only, but anyways, that's not the concern. The concern is, why are you talking about this, Jasmine ma'am? The reason that I'm talking about this is because this portion of the filament which attaches to the thalamus has a name. It's known as the proximal end. It is known as the proximal end. Proximal means nearer end. Okay. So, ma'am, what do you call this portion which is going to attach to the anther? This portion, since it is a distance, I will call this as the distal end. So there is proximal end and there is distal end. So if the question comes, where on the filament is the anther located? The proximal end or the distal end? What is your answer for that? In the distal end you have. Why? Because we want to make sure that the anther is at the distance such that the wind can take away all the pollen grains. In that way you can remember, don't mug it up. Okay, so this is the basic logic that we have. This is the only thing that you need to know from this particular portion. Now we are going to go ahead and see what is going to be there inside the anther, because that is super important. So anther, I have just zoomed it in and it looks like this. So if you also just look at it once, okay, just look at this papa guy out there. What do you see? Ma'am, I see there's something in middle dividing it into two. Okay, two what? I can say two lobes. I can say it is two lobes. Okay, so there is this one lobe and this is another lobe. This is one lobe and this is another lobe. Okay, what do you call this condition of anther where you are divided into two lobes? It is known as bilobed condition. Bilobed means bi means two and lobe means lobe. Basically parts, right? Now there is something that you cannot see but I can see because of the brightness, and I'll show that with the help of a diagram. So there is something that is dividing the lobe also. This is the lobe. The lobe is also divided into two and you cannot see it. Why? Because there is too much brightness here and there. So this division that is going to happen within a lobe, what do you call this as? What do you call this as? So basically it looks something like this. This is your lobe, this is another lobe. The lobe is also having division like this. And what do you call this as? The divisions, you call them as theca. What do you call it? You call one of this as theca. So this one portion is one theca. This another portion is going to be another theca. So how many theca does one lobe has? This is one theca, this is another theca, and this is one lobe. This is one lobe. So can I say one lobe is dithecous in nature? Can I say that it is dithecous in nature? What do you mean by di? Basically two theca are there. So again I'll say that this is a bilobed structure anther, and not just that, I'll say anther is basically having a dithecous nature. It is having what? A dithecous nature. That means one lobe, whichever is there, both of them are going to have two theca for themselves. This one also has, that one also has. Okay. Now ma'am, I understood that. Okay, I understood that and I feel it's boring, but it's so okay. Let me still believe in you and let me go ahead. Okay. So when you go ahead by believing me, I am going to give you some exciting news. Let's do one very important thing. This is just an outside thing of how things basically are appearing to us. We need to go within and check what's there inside the anther. For that, we have to do a thing that is cut it up. Cut it up and then do what? Cut it up and then look it from the top. Just imagine you are having this, you are cutting this and basically looking at it from the top. What is the view that you see? This is the view that you're gonna see. Ma'am, I still don't get it. How can we get this view? So listen to me, I'm going to explain about that, and basically this picture is actually very important. In your NCERT you have some similar picture like this about the walls of the anther and things like that. So for understanding that, you have to understand this. Before that, I missed a point which I have to mention, and that is, I told you about this line that goes in between, right? What do you call it? Longitudinal groove. You call this as the longitudinal groove. Longitudinal means it's very long, okay, and groove means basically it is within, it's within, okay, there's a depth in it, in that particular groove. So longitudinal groove is there, dividing the lobe into two theca. Now basically I was talking about this. When I cut it from like this and look it from the top, it is going to be appearing to me something like this. And if you're wondering how, so listen, this is going to be the one lobe, first lobe, and this is your second lobe. Okay, and I told you each lobe is divided into two theca. So this is your one theca This is your second theca. Similarly, this is this lob's first theca, and this is going to be the second theca, which is exactly why you are seeing a picture like this. So this is one lobe, this is another lobe. In here you see this is one theca, this is another theca, this is one theca, and this is another theca. In between that you see something like this, like this, like this, like this. What is this? This is known as microsporangia, and this is where the pollen grains are going to be formed. I told you in the anther you have pollen grains, right? So where are the pollen grains formed? In these things that you see over here. What do you call this as? Microsporangia. Ma'am, do you have any other name for it, or do they become something else later on? Yes, I'll say that after some point they become pollen sacs. Why pollen sacs? Baba, because they are going to form the pollen grains. Until now there are some things happening over here. They have not started making pollen grains. Soon they are going to make. There are processes that is going to happen. In that case, what will you call now the microsporangia as? You can call that as the pollen sac. Why? Because the pollen grains are being made there. Got it? So microsporangia, pollen sac is considered to be equal. The same thing, synonymous things which are there. And how many microsporangia do you have? In total we have 4 of them. In total we have 4 of them. Inside that there will be so many pollen grains that are going to be formed. But how many of them? 1, 2, 3, 4. In each theca, how many of them will be there? In each theca, only one. In one theca, 1, in another theca, 1, in another theca, 1, in another theca, 1. In each lobe, how many will be there? This is one lobe, it has 2. This is one lobe, this also has 2. Okay, so do understand microsporangia are located in each corner of each theca out there. Basically in that case, I'll say one lobe has 2 theca. That means one lobe will be having 2 microsporangia as well. Okay, done. Now we are going to go ahead and see what's inside there. But before I do that, let's read what is written over here such that everything is sorted up. Now, anther is bilobed with each lobe having 2 theca, that is they are dithecous. Anther is bilobed and each lobe has 2 theca, which is why they are known as what? Dithecous condition. Studied. A longitudinal groove runs lengthwise separating the theca. Lengthwise like this. Okay, separating both the theca out there. Okay, that is also done. Anther is 4-sided tetragonal structure. Tetra means 4, okay, and it has 4 sides, 1, 2, 3, 4. I spoke about it. Yes, that is also done. Consisting of 4 microsporangia located at the corners, 2 in each lobe. So 2 in each lobe you have it, and basically in one corner you have 1. In total, how many you have? You have 4. In the KCET question paper, if at all they are asking you in each corner how many you have to say 1. If they are asking you in corners how many you have, basically talking about all the corners together, so in that case say 4. Okay, yes. Now the microsporangia develop further and become the pollen sac. I told you they are the ones that is going to become the pollen sac later on. That is also clear. Yes, ma'am. Done. Okay. Now we are going to see the wall layers of basically the microsporangium, or you can also say the anther wall layers. Okay, the anther wall layers. So for that, this is the picture which is there, but I want to go in depth and I want to explain you, which is why I'll make my own picture. And by the way, everyone who have not hit on the subscribe button yet, what are you waiting for? Hit on it right now and do let me know if at all this video was helpful for you or not. Okay, so over here you have the anther. Come on, work hard. It goes like this. It goes like this. This is Chutko's anther. Okay, now I know that looks bad, but it's okay. We have the microsporangia being made over here. Let's make it. Okay, first and the foremost thing, inside this, what do you have? Inside this you have so many cells out there. And when cells come together, what do you call this as? You will call this as a tissue. When cells come together, you can call this as a tissue. So I have a name for it. I'll call this as sporogenous tissue. Sporogenous tissue. What is the tissue made up of? Tissue is made up of cells. Made of cells. What do you call the cells as? You call the cell as pollen mother cell. Why? Because they are the ones that are going to form the pollen grains. So they are the mothers of the pollen grains. So can I say they are known as pollen mother cell? Yes, ma'am. Obviously, you can say whatever you want. So over here, this is what? The pollen mother cell. Okay? This short form, you can call it as PMC. But But ma'am, in our NCERT something else is written. It is also known as microspore mother cell. It is known as pollen mother cell, that is there, but they have also mentioned that it is also known as microspore mother cell. Is that true? Yeah, that is also true. Microspore mother cell. But how can that be? How can that be? You are saying that it's the mother of the pollen grains, and now you are saying it's the mother of the microspore, something that is known as microspore. Yes, and I'll explain that to you in the next portion, probably in the next slide up there. So wait and be curious, okay? Be curious to know about this, because I want that curiosity. Okay, now we are leaving them like that only. So this is what? Just a tissue that is known as sporogenous tissue having the pollen mother cell or the microspore mother cell. Okay, done. Now what? Obviously, if at all they are going to do all of it and going to give rise to pollen grains, there should be someone to nourish them. Obviously, they cannot sustain by themselves, which is why they have cells around it. Okay, they are going to have cells around it. And what do you call the cells as? You call this as tapetum. Or basically this particular layer which is there, you call this as tapetum. And you know one very unique thing about tapetum? Okay, tapetum wants to nourish these cells out there, which is why they say that if this is a cell, the nucleus of this particular cell will be dividing into two. If this is a cell and it has one nucleus, the nucleus is going to divide into two. Why? Expecting that the cell will also divide into two. But it does a cheating. It says that, okay, I have divided the nucleus, that is karyokinesis has taken place. Karyokinesis means division of nucleus. That has taken place. But cytokinesis, cell division, I will not allow it. Why? Because I need one big cell having enough amount of cytoplasm, because this cytoplasm will be having the nutrients out there which is going to nourish the pollen grains later on. So can I have a little more cytoplasm and can I not undergo cell division? So the cell says, okay, fine, you can do that, since anyway you are the one who is going to nourish it. Which is why you need to understand that tapetum, it is made up of cells which has dense cytoplasm. And apart from dense cytoplasm, there is one very important thing that you need to remember, and that is they are going to be binucleate. Binucleate means they are going to have two nucleus. Okay, they are going to have two nucleus out there. Why this happened? Because of failure of cytokinesis. Cyto means cell, kinesis means division, cell division. Done. Okay, ma'am. Fine, then that is done. Okay, now what? So this is one layer, tapetum, which is actually the fourth layer. So ma'am, you are saying there are three more other layers that I need to remember? Yes. And we'll start with the outermost one. So over here, the outermost layer that we have, just a second. So whenever I change the color, it stops. The outermost layer that we have is known as epidermis. Epidermis. This is the first one. Okay, inside the outermost layer, you have the next layer out there. What do I call this as? This is known as endothecium. Endothecium. Okay, ma'am. Then do you have something else? Yes. This space that is left behind over here is also filled of so many cells, and basically this layer that is going to be formed, is it visible? Slightly less, so I'll just change the color. So over here, this is known as the middle layer. Middle layer. Ma'am, is this diagram important? Yes. You could be asked to make this diagram, so make sure you know about this. So epidermis is there, endothecium is there, middle layer is there, and the fourth layer is your tapetum. And how do Jasmine Mam remember it? Jasmine Mam remembers it with the help of this. EMT. You have went to ENT doctor. I have said this so many times to so many children that they were like, oh my god, ma'am does not know anything, any other clue. But if at all you have any other trick to remember this, please put it down in the comment section so that I can probably upgrade myself. So this is ENT doctor, ear, nose, throat, right? So similarly over here you have EMT. Okay, epidermis, endothecium, middle layer and tapetum. Trust me, this is going to help you in your case set, because they will be asking you in which order is it placed, so you should be knowing that. And not just that, the next question that could be coming in second PUC, as I said, is a diagram, and the next question coming in the second PUC could be, or in case it also could be this, that is, which layer helps in nourishment? This layer, we know that this is there for nourishment. Okay, then what about these three layers? Papa, guys, chumma are waiting or what? No, Baba. They are very, very important for two very important things. Okay. Two very important things. And what is the use of that? First one is going to be for protection, obviously. Protection. Second one is going to be for dehiscence of anther. What do you mean by dehiscence of anther, ma'am? Dehiscence of anther means breakdown of anther. Ma'am, why are you breaking down anther? Such a bad person you are. So listen to me, pollen grains are made over here inside. No, these guys have to be released. How will pollination happen then? So for that, dehiscence of anther has to take place. Now if these guys are not ready to break open, do you think the pollen grains will be out? No. Which is why you need to know that yes, they help in protection. The second thing is that they help in dehiscence of anther also, breakdown of anther, such that basically the pollen grains can be released. So these are the two functions of these three layers. This is the function of this layer and this is the feature of this layer, and I hope all of it is being done. Okay, so I'm just repeating. You have EEMT, epidermis, endothelium, middle layer, and tapetum. Tapetum cells, which is there, it is there for nourishment, which is why it has dense cytoplasm and two nucleus. Why two nucleus? Because cell division did not take place, okay, but nuclear division took place. That is the exact reason that we have. Okay, now what's inside it? Sporogenous tissue. What is it made up of? Cells. What cells? Pollen mother cell or microspore mother cell. Done? Done. Okay. Cool. Now we are going to talk about what happens with this and why do you call this as microspore mother cell as well. By the way, children, if you have any doubt, do let me know. Please know. Okay, so there's a question. Consider the following statement and choose the correct answer from the given option. So, statement number 1 says, innermost layer of microsporangium is tapetum. Is it tapetum? This is 2021 question of KCET. Innermost layer of microsporangium is tapetum. Obviously, yes. The fourth layer, yes. Cells of tapetum possess dense cytoplasm, more than one nucleus, and nourishes the developing pollen grains. It has dense cytoplasm, and we know it is binucleate in condition, more than one nucleus, and it nourishes the developing pollen grains. So, can I say both the statements are correct? Yes, ma'am. Statement 1 and 2 are correct. Do I read all the options carefully? Yes, I do read that. And after that, I come up with the conclusion that B is the answer for this particular question. Both statement 1 and 2 are going to be correct. Okay, yes, done. Now this is how we're gonna go, and I hope you like the pace of it. If you feel that I'm very fast, you can slow it down at the corner. If you feel that I'm very slow, you can speed it up. Okay, and one more very important thing, by the way, if you're not aware of this, so basically, you know, we have come up with all in one page, and if you have not seen, there are one shots released for mathematics as well. Okay, on relations and functions, there is one shot that Sir did. Okay, Somik Sir did for another chapter, which is electric charges and fields. So basically, all of it, how can I say it together? If you go check out this video, or basically probably under my video, this particular video, there will be a link. Let's check it out. So there's a link. Access the course over here. This is nothing. So if you're not able to see, I can zoom it in. Access the course over here. If you click on this, basically you will come to this particular page where you do not have to buy anything. It is of 0 rupees. Okay, you would have to fill in your details, after which you will be able to see all these subjects at one place. So that means whatever is provided in YouTube, and if you're not able to find it between the chaos of counseling and this and that, you will be able to see it there subject-wise allocated. What all concepts are being done, what all chapters are being done. In biology, it will be all together. What all things are being done in, mathematics, all of it will be there. So make sure you check it out. This is again a free thing which is there. You have got such features where you can ask, doubt and all. There is Insta Solve and stuff like that. Make sure you go ahead and watch it. It is definitely going to help you guys out. Okay, it's known as all in one page. So that's a free thing. If you want to go ahead and check it out, you can. If you do not want, then it's okay. Okay, that's all your wish. Fine. Now coming back to this, that is all about microsporogenesis. What? So basically we spoke over here that we are going to call this as microspore mother cell, and we also call this as pollen mother cell. The reason is that I'm going to break the suspense. The reason is that actually the cell which is there, this particular cell which is there, it initially divides to form what? To form something that is actually known as, come on, Jim Brumbra, yes. So it is actually known as microspore. This microspore later on transforms or basically forms into pollen grains. It is going to form into into what? The pollen grain. That is how things are going to go. That is why you call this as microspore mother cell, because it forms the microspore first, and then this microspores later are going to help in the formation of pollen grain, which is why we can generally say that anyway at the end you get pollen grains, so it is known as pollen mother cell also. But initially you have to form the microspores, and that process of formation of microspore is known as what, Baba? It is known as microsporogenesis. It is known as microsporogenesis. Now later in this chapter you're going to see megasporogenesis in the case of female. How do you remember it? So usually remember in the case of human beings also, the female gamete ovum is big, but the male gamete sperm, it is microscopic, very small. So micro, male gamete small. Okay, male gamete micro. So you remember like that. This for the formation of male gamete later on. Okay, it's not that basically formation of male gamete, but anyways, male process includes what? Microsporogenesis. Microsporogenesis is the formation of what? Microspores, and we are going to see that right now. So what exactly happens over here? You have the microspore mother cell. Okay, you have the microspore mother cell, and I'm clearly understanding you're not able to see, so I'll just lower down the brightness a bit. And then I think things could be better. Yes, this could be far better. Yeah. So we have the microspore mother cell. This microspore mother cell is going to undergo division. What division, Baba? It is known as meiosis. Meiosis. Listen to me very carefully. Whenever I am planning to form gametes at the end, I have to make sure that the gamete carry only half the number of chromosome. Why? So, just giving you a situation. Okay, just giving you a situation. So over here there is Pappu and this is Puppy. Both of them are there. Okay, now they are planning to have a baby. They are planning to have a baby. What is the cell in them? The cells which are there, how many number of chromosome do they carry? 46, ma'am. Here also 46. All human beings have 46 number of chromosome. Okay, fine, got it. What if the gametes which are there, that is the sperm over here and the egg over here, what if they also carry 46 number of chromosome? What is going to happen? Baba, the sperm and the egg will combine together one day. It becomes what? 92. Is 92 a normal number of chromosome that we have? No, that should be 46. So then how can we make it possible? This is not at all possible. Same thing applies over here, Baba. In plants also, it cannot be possible. We have to maintain the number over here, this particular number, that is what was there in the parent plant or the parent organisms out there. So what do? We make it undergo a division where if a cell has 46 number of chromosome, at the end it becomes half. That is what? It becomes 23. What's the use of that then? Now you suppose over here, if it is 23, okay, just a second. To have a clear idea, if it is 23 and if this is also 23, if it is having only half the number of chromosome, sperm, okay, and egg is also carrying only half the number of chromosome, when this and this half comes together, it becomes 46. Isn't it like the normal cell? Yes. So that is the use of it, and that's how the heart is being made. Anyways, so this is what I was trying to say. Whenever you are forming the gamete, there's a division takes place, and it is known as meiosis. It is known as meiosis. Okay, where it is also known as reductional division. Why do you call it as reductional division? Because you want to make sure that the number of chromosome is reduced into half. Then this half of a half can come together and make a full number of chromosome later on. And meiosis always happens in two stages. Okay, so if this is a cell, this cell divides into two first. Then this cell, okay, this is first division that has taken place over here. Now what? Again, this cell which is there will be dividing into two. This is what? The second division that takes place. Every meiosis, when I say meiosis is happening, it happens in two stages completely, 1 and 2. Now listen to me very carefully. I told you at the end we want 23 number of chromosome. So at the end of first division itself, the number of chromosome actually becomes reduced. The first part of meiosis itself, the number of chromosome is going to be reduced, and then the second part of the meiosis, will it reduce again? No, we cannot make it more half. It remains the same. it remains the same. Why do you call it reductional division? Because in the first one itself, it has reduced the number of chromosome. And at the end product, anyway, we have the reduced number of chromosome when you compare with this, right? Right, ma'am. That is the logic. So always remember, if you want to form the gametes or something related to the gametes, please make it undergo meiosis. And in technical terms, we are going to use a term that is known as ploidy. Okay, if we have double the number of chromosome, we call it as diploid. What do we call it? Diploid. So over here, it actually has, this is double the number of chromosome, 46 is double the number of chromosome. So I'll say it is what? Diploid. And how is it represented? It is represented by this, 2n. Okay, n is there, now it is having double, which is why you call it 2n. So ma'am, in that case, what will I call this? It has only half of it. So what do I say? So basically half of 2n. It becomes what? It becomes n. This is n. Okay, n. But what do you call this as? Here you call this as diploid, right? What do you call this? Half of diploid is haploid. This is known as haploid. So if you are asked what is the ploidy of the microspore mother cell, you will say diploid. What is the ploidy of the microspores which are going to be formed? They are going to be haploid. Remember that. Cool? Cool. So this is 46. This is about human beings, I've said. But anyway, this is the logic that you want to apply. That is why I just gave you an example. Now over here, when we come back to this, you do not have to remember the number of chromosomes and all. You just have to know, if this is 2n in nature, microspore mother cell, then at the end of meiosis, you're going to get two cells which are going to be what? N and n in nature, which means they are going to be haploid. And this we started with what? Diploid. Cool. Now this cell again divides because meiosis happens in two stages. This cell divides into two. It remains the same. It remains haploid itself. What do you call all of them? You call all of them as microspore. What is the thing about microspore that we need to remember, ma'am? That microspores are like us, always together. Okay, always together. So these microspores, Baba, you know what they form? They all come together to form something that is this, that is known as microspore tetrad. Tetrad means 4 of them together. Okay, so we have microspore tetrad being formed over here, and you need to remember it. Okay, ma'am, why are we talking about all this? We wanted to make pollen grain know what happened there. So listen to me. This microspore tetrad, when they are formed, they're all together. We have to do one sad thing. We have to make them do breakup. Everyone has to do breakup. Okay, so for that, the condition in the anther has to change. Everything is happening inside the anther only, right? The condition in the anther has to change. There has to be dehydration. So when there is dehydration, when the water content is going to be removed, the microspores which are like this, they're going to move away. Oh my God, they're gonna move away. And when I say that they are separated, we say that they will further undergo division and form what? Your pollen grain. And form the pollen grain. So each microspore tetrad which is there, Baba, it is going to dissociate from each other, move away from each other when there is dehydration, and finally they are going to form the pollen grain. They will be forming the pollen grain. Ma'am, how many pollen grain? How many microspore do you see? 4 of them. So 4 pollen grains are going to be formed. 4 pollen grains are going to be formed. Clear? This process from here to here, actually from here to here, until the formation of microspore, is known as microsporogenesis. Later on, obviously, it helps in the formation of pollen grain as well. Mention that. So sporogenous tissue, each can form a microspore tetrad. Each cell in the sporogenous tissue. Okay, so my sporogenous cells is what you call them. Basically, you can call them pollen mother cell, microspore mother cell. That will be more easy. So I'll just remove this and I'll write it as, okay, PMC or MMC. Microsporogenesis is what? Meiosis in pollen mother cell produces microspore. This is known as microsporogenesis. Now it can be pollen mother cell or it can be microspore mother cell. Now microspore tetrad initially grouped in 4, but later they separate. So at the end you get microspore tetrad. Initially they are grouped in 4, but later they are separating. Why? Because the anther is maturing. When the anther mature, the anther is going to dehydrate. The microspores dissociate and develop into what? The pollen grains. And ta-da, we have got it. Pollen release. Thousands of pollen grains are released when anther dehisces. So when anther breaks open, so many, so many, pollen grains are going to be released. That is the basic logic that we need to remember. Cool, ma'am. Done, ma'am. Okay. Then in that particular case, we have to go ahead and solve a question. In a diathecous anther, each pollen sac contains 1000 mmC. What is the total number of pollen grains produced by the anther? Did you understand anything? In a diathecous anther means so many anthers? No, normal anther is diathecous, no. So we're just talking about one anther. Whenever you get something like this, make a picture of it and that's how you remember it. So in a diathecous anther, each pollen sac contains how many mmC? 1000 mmC. So over here, pollen sac is there, and how many mmC are there? 1000. So write it, 1000. Here also 1000. Here also 1000. We have 1000 mmC being formed. Okay. Now what is the total number of pollen grains produced by the anther? How do I know that, ma'am? So 1 mmC which was there was producing how many microspore at the end? Ma'am, it was making 4 microspore at the end, 4 of them were making pollen grains. So if there are 1000 will also, each of them are going to give rise to 4, right? Each of them will be giving rise to 4 of them. So 1000 into 4, what do I get? 1000 into 4 is what? 4000, ma'am. Here also 4000, here also 4000 pollen grains, here also 4000 pollen grains. So can I say 4000 into 4? It becomes what? 16,000. Listen, we had 1000, we had 1000, we had 1000, we had 1000. Each of them produces 4 pollen grains. So here 4000, here also 4000, here also 4000, here also 4000. So 4000 into 4 becomes what? 16,000. Isn't that the question? What is the total number of pollen grains produced by the anther? It is going to be what? 16,000. Got it? This is 2016 question that have come in KCET. Works? Works, ma'am. Works perfect for us. Now we're going to go ahead. I hope until this particular point it's clear to all of you. Clear? Right? Everyone who is watching this, do you have anything to talk about it? Well, by the way, since I remember about it right now, let me talk about it. So there's a course that we have. It's a paid course. It's known as Pariksha Veera. Okay, it's known as Pariksha Veera, and basically this is for all the children who are preparing for second PUC and KCET seriously. Now you might find if someone from there is there, probably over here, I'm saying that, ma'am, we have studied everything. So, or basically I can make it easy for you. If you're probably watching this video and if you feel that, okay, it would have been really good if at all there was ma'am teaching to me in live manner, and there were basically all the questions of KCET were solved, and not just that, everything, each and every doubt was being catered and things like that. It would have been better. It would have been even better. So this one shot is definitely helping me out, but in this particular case, I can interact more to her and basically get to know more about things, and that is going to be really good, right? So if you're someone who is wondering like that, please know that we have already launched the Pariksha Veera course. The classes have already begun. And we are going to make sure that the second PUC is going to be completed by the month of November, and there's an exact date also. And what is the date? Date is November 6th. Okay, the pricing for this is 3,499, and it's not just about this. There will be 20 plus mock test, part and full length, all of it which is going to be provided there. There will be unlimited DPP generators which children can use to generate their DPPs or the daily practice paper and solve their question. And this is available not in YouTube but in a separate app, that is Vedantu application. Okay, so if you're someone who have not checked it out, make sure you check it out. The link will be there in the description section of this particular video, and I'm pretty sure it is going to help you out. If you think that this video had helped you out and you need a proper one to, and it's not like it's just a live, it's like every schedule is being fixed. It is going to be a fixed schedule that on this day this topic is going to be covered. You have 2 classes a day, one for one subject, another for another subject. Each class is of 1 hour 45 minutes, and obviously the depth is going to be more. I am pretty sure that child who probably have attended my classes in Pariksha Veera will remember, okay, will be able to recollect each and every question that I have discussed out there and could tell the answer like this also. Though that is something that I clearly am sure about. So if at all you have not checked it out, probably go and check it out. And just give me a second. Now I'll increase the brightness and come back again, or it will be too dark. Okay, cool, works. So now we're going to go ahead and talk about the male gametophyte. What do you mean by male gametophyte? The one that is going to form the gamete. It's not the male gamete. I'm talking about the one who is going to form the gamete. Ma'am, who is that? Until now, we started studying about the male reproduction. Until now, you have not told that who is the gamete. We were assuming that pollen grain is the gamete. Array, pollen grain has the gamete, which is why the male gametophyte is none other than the pollen grain. It is none other than the pollen grain. Pollen grain is what is known as the male gametophyte. Remember, this is a mistake that we all do. Pollen grain is the gamete. No, it forms the gamete, and we're going to see how it does. And please excuse me, let me drink some water. Very, very thirsty. If you have not had, please have one bit of water. Okay. Now, we're going to go ahead and talk about the structure of pollen grain. We got the pollen grain from where? From the microspore. So, you need to understand, we had the microspore over here. Suppose this is the microspore. Microspore with the nucleus that is haploid, right? This microspore underwent a division. Okay? It basically underwent a division that is known as mitosis. Or, yeah, that is the best way to remember it. And that's how you got your very, very important cell that is known as pollen grain. So I'm just making it over here. This is going to be your pollen grain. When I say that this underwent mitosis, that means a cell is going to divide into two. What is mitosis? In mitosis, the cells divides into two and the number of chromosome remains the same. For example, we were over here when we were having 23 number of chromosome, right? So when it divides, will it reduce? No, it remains 23 only. This is known as mitosis, and this is something that happens normally in all the cells. Cell division that happens in normally all the cells. In gamete, it is meiosis. So here, we are actually forming the gamete, but before that we have already underwent meiosis, which is why we are not going to undergo meiosis once again. We're just going to maintain this ploidy. We just want to make it half. We had made it half. Now we're going to just maintain the ploidy, which is why we're just going to make it undergo mitosis. When it undergoes mitosis, Baba, you get two cells. This is what I am talking about, a pollen grain. A pollen grain, and it has two cells. One cell over here is a very big cell. As you can see, this is a very big cell. Inside the cytoplasm of this very big cell lies a very small cell. Okay, so this very big cell, first of all, let me call it a name. This is known as vegetative cell. It's known as what? Vegetative cell. And it has a nucleus. Ma'am, this is not a nucleus, this is amoeba. Yeah, exactly. That's what I wanted to say. It has an irregularly shaped nucleus. Ma'am, is this important? Yes, came in your second PUC. It has an irregularly shaped nucleus. Okay, ma'am. Done, ma'am. Cool, ma'am. Got it, ma'am. What about this cell that you were talking about? This is known as generative cell. Generative cell. Okay, what is the use of generative cell, ma'am? They're going to generate something. What? Electricity? No, they gametes. So generative cell, Baba, they will undergo mitosis again. They will undergo mitosis again to form what? One gamete and the second gamete. So as of now, it is like this. One cell is there with what? With elongated nucleus. So this is a spindle shaped cell with an elongated nucleus. The shape of the cell is spindle shaped, but the shape of the nucleus is what? Elongated. Please remember this. The shape of the nucleus has all come. That's why I'm telling you. Okay. Later on, when I said that they are going to undergo mitosis, what exactly happens is that this vegetative cell remains like that. This cell which is there, it divides into two. This become one gamete and this become another gamete. Okay, so pollen grain can be like this where they have three cells. It can be like this where they have three cells, and it can be like this where they just have two cells, one this and another this. Okay, that is the scenario that we have. Mostly when I say a pollen grain is being shed, it is being shed, that is when anther-dehisces and the pollen grains are released, it happens in two cell stage only. Okay, it will be having just two cells. So I'll say 60% of angiosperms, as in flowering plants, they are going to shed the pollen grains in two cell stage. Okay, what about the 40%, ma'am? The 40% are going to shed in three cell stage. Okay, so majority in two cell, little bit in three cells. Okay, only few of us are there who are multi-talented, right? So in that way you can remember, only few of us are there basically who are going to have many cells out there, multi, okay? So that is the basic logic that we have. This is very important, this particular portion, which is why you need to remember it. Okay. Now we're coming back to this. I just have 2 cells right now, and I'm going to talk more about it. So this cell, you know, it's going to form the gamete. I got it. But why do you have this very big cell as? You know, very big cell has dense cytoplasm, and when I say dense cytoplasm, you should understand it would be for nourishment. Nourishment of what? Nourishment of this particular thing which is there. So vegetative cell which is there, it is important, very, very important for nourishment. Of what? Of the generative cell which is going to lead to formation of the gametes out there. Okay, ma'am, done. What apart from it? So this cell, obviously a cell has a plasma membrane. So I'll just say this is the plasma membrane. Okay, ma'am, got it. This is the plasma membrane. What else? After the plasma membrane, the second thing that you have to remember is that they have a continuous layer around it. Remember, continuous. I'm using the term continuous. Okay, what do you mean by continuous? That means there is no bit break in between. It's very, very smooth and it's together. There is no small pore in between. This particular thing which is there is known as intine. It is known as intine. Intine means it is inside something, it is internal layer. And you know what it is made up of? It is made up of Priyanka Chopra. What, ma'am? Priyanka Chopra. Yes, Priyanka Chopra. So always remember Priyanka Chopra. Pectin and cellulose. Pectin and cellulose. These are the 2 things that are going to make up your intine, and always remember this particular intine which is there, it is going to be continuous in nature. It is going to be continuous in nature. Got it, ma'am. Okay, works, ma'am. What apart from it? So there is another layer that is there. So this is one layer, intine. Apart from it, you have another layer. Ayyo, where is it? Yeah, this layer. And if you notice what I'm doing, it's not continuous. It is going to be what? Discontinuous. It is going to be discontinuous. And what do you call this layer as? This layer is your exine layer. Exine means the outer layer that is there. Okay, ma'am, and what is it made up of? Here it is made up of Priyanka Chopra, pectin and cellulose. What is it made up of? It is made up of the most toughest resistant material out there, which is known as sporopollenin. It is known as sporopollenin. Remember, this is one very, very important thing. Why pollen grains can be preserved as fossil. Okay, it can remain unchanged for so long. Why? Because it is made up of the outermost layer is highly resistant. It can resist alkali, acid, anything. And what is that thing? It is basically sporopollenin that makes up this particular exine. Okay, ma'am, got it. But why do you have pore over here? Basically, these pores which are there, they are the area where this particular guy is going to be absent. Where sporopollen is going to be absent, that's where you have the pore, and this pore is something that you know call as germ pore. What do you call this as? Germ pore. Why is germ pore important for you? Germ pore is going to be super important. The reason being that, you know, this is a pollen grain and basically it is going to form the gamete and stuff like that. When the pollen grains go to the female organ, the female reproductive part, basically from air they land up on the female reproductive part. The female gamete which is there, it is very inside, and basically this male gamete has to travel, and for that it has to form a tube-like structure. It has to form a tube-like structure which you call as pollen tube. This pollen tube, where will it come from? Basically there should be some space for it to come, no? For that you have the germ pore. So whenever the pollen tube is going to be formed, it basically comes through that particular thing. So for your imagination, I can make it something like this. It will be coming something like this, where you have the vegetative cell over here and you have the gametes, right? So this is one gamete, this is another gamete. The gamete will be traveling down through this. Okay, got it. Ma'am, so, but I didn't get it how pollen tube is going to be formed. Who helps in the pollen tube formation? Listen, that's the next role of vegetative cell. This very big cell is nourishing it. Apart from it, it helps in the formation of the pollen tube also. It provides content, cytoplasm and all, for the formation of pollen tube such that the male gametes could travel. And the area where it goes through would be the germ pore. So remember, vegetative cell which is there, it is there for nourishment. And the next thing is that it helps in the formation of pollen tube, which is why it is also known as tube cell. which is why it is also known as what? Tube cell. Tube cell. Now this is not there in your NCERT. Somewhere it's there, somewhere it's not there. I mean, it's not actually there, but sometimes they could be asking you questions, not direct questions related to it, but I have seen a question in KCET that has come from this topic, which is why I'm telling you, please remember it. Okay, so to summarize everything that we studied over here about pollen grain, which is what? The male gametophyte. First of all, it is known as male gametophyte. Why? Because it carries the male gamete, it forms the male gamete. How does it form? So microspore is there, it undergoes mitosis, you get 2 cells. Again, there is next mitosis, that is this, where you get the gametes. So always understand, if at all you are talking about pollen grain formation, okay, if you're talking about pollen grain formation or basically the gamete formation, anyway, you have 2 mitosis there. This is the first mitosis, this is the second mitosis at the end of it, where you get 2 gametes. Okay, at the end of first mitosis you get 2 cells, at the end of the next mitosis you get 3 cells. Remember it. If you want to form the gametes, you have to do 2 mitoses, one here, one inside. Cool? Yes. Now here you have 2 cells, vegetative cell and generative cell. Vegetative cell has dense cytoplasm, irregular nucleus, and this vegetative cell is dense. Why? Because it wants to provide nourishment to the generative cell. Why nourishment to generative cell? Because generative cell is the one that is going to form what? Is going to form the gametes. How many gametes, ma'am? 2 gametes. Okay, ma'am, so pollen grains are usually shed in this 2 cell stage? No. 60% of them, yes, they do that. 60% of them are going to shed their pollen grains in 2 cell stage, but 40% of them, very less number, they are going to be having multiple cells. Basically, this generative cell divides into 2, and hence in total we say this 1 and 2 gametes, basically 3 cell stage. Got it? Got it. Now we have a plasma membrane around it. Around it we have intine. Intine is made up of cellulose and pectin. Pectin and cellulose, either way. This is continuous, this is not discontinuous. Please remember, questions can be coming related to that. Now the outermost layer is discontinuous. This is known as exine. Exine is made up of sporopollen. Sporopollen is the most toughest resistant material out there. It can resist anything, alkali, acid, anything, which is why pollen grains can be stored in the form of fossils as well. Got it, ma'am. Cool. That is also clear to me. Everything is clear to me. Just one thing. What is the diameter? Does it have any diameter and all? Yes. So basically, I'll say it is almost 25 to 50 mu meter. Mu meter means, mu over here means micrometer. Okay, it is 25 to 50 micrometer. Remember that also. And that's all. Okay, so let's see. So over here, what do we have? Male gametophyte, the pollen grains represent the male reproductive units, and that is the male gametophyte. Observation, variety in shape, size, color and design under the microscope. So there are definitely different types of, pollen grains are there. Size is what? Spherical. 25 to 50 mu meter is the diameter of it, is the diameter. Wall layers, exine, that is outer layer, made up of sporopollen. And what else? What do you have over here? You have one more thing, that is intine, the inner layer, which is made up of cellulose and pectin, or pectin and cellulose. Cytoplasm is there, enclosed by plasma membrane. Yes. Cell types is there, 2 cells. One is vegetative, another is generative. Vegetative one is going to be larger. It stores food and has an irregular nucleus. Okay. Generative cell is going to be smaller, spindle shaped, and floats in the cytoplasm. Yeah, it is having the vegetative cell has a bigger cytoplasm. No. So basically, we say the generative cell floats in the cytoplasm of the very big cell. Okay, the very big cell. So V for very big cell. Remember like that. It will be very easy for you. And remember this, it has elongated nucleus. Not spindle shaped nucleus, elongated nucleus. Okay. Now the pollen shedding. 60% of the plants shed at 2 cell stage, 1 vegetative and 1 generative. 40% of them are going to shed at 3 cell stage. 1 generative divides to form 2 gametes as well. That is why 3 cells in total. 1 vegetative and 2 gametes. Done. Now this is how it actually looks. If you see, this is how it actually looks. So this is a tube cell. Tube cell means vegetative cell. And this is the generative cell. Small thing, small little thingy that you see over here with the nucleus. So in this does not look like elongated nucleus and all. Obviously, everything in NCERT could not be exactly correct in all terms. Exceptions could be there. But yes, got it. So this is about it. And I really want to, take up a question over here. So this is an MQP, model question paper question of previous year, okay, which said, write the significance of sporopollenin. So how do you write it? Sporopollenin is one of the most resistant substances forming the exine of the pollen grain and protecting them from high temperature, strong chemicals, UV radiations, and also microbial attack. Different types of problems that are there in the life, it sorts everything. Because of its extreme durability, pollen grains which stand with sporopollen gets preserved as what? As fossils also. They can be easily preserved as fossils as well. This is how you're going to frame your answer, and always write your answer in points. Cool. Next is, draw a label diagram of mature 2-cell pollen grain. This diagram, do I need to draw it? No. It's okay if at all you do not draw that. You can draw this one. Teachers are gonna understand. Teachers do not write 25 into 50 over here. That's okay. Rest everything you can draw like this. Okay. I hope that works. Please make sure you have a practice of it. It's very, very important. Label them, and when you label, do not label it like, what do you say, in all the direction, okay, like a cross. Rather, make sure you put it in one direction, either this or this. Or if you want to do in both, make sure you put a straight line and draw it. That will be really, really good. Cool? Yes. Now, next, which of the options is true in context of above diagram of pollen grain? Now, this is a question that have come in KCET 2025. Okay, 2025, very recent one. So, over here, see. When you get this type of questions, first of all, this is given as A, this is given as B. Please mark what is A and what is B, and not in your head, you have to write it over here. So A is the very big cell, we can see that, and this is the small cell, generative cell. So let's write it. A is vegetative, B is generative. First option, A is generative, wrong, because we know that A is vegetative. Second option, A is vegetative. Okay, we'll keep it there. Third option, A is generative. We are going to cross it up again because A is not generative. We know it's vegetative. Okay. Now we just have this and this. Eliminated the options that we do not want. Now we're going to check your answer. A is vegetative cell which gives rise to male gametes. Vegetative cell is very big cell, helps in nourishment, not giving rise to the male gametes. So this part is wrong. Again, wrong question. A is vegetative cell with abundant food reserve, and B is generative cell which forms the male gamete. Right? B, generative cell, forms the male gamete. Vegetative cell helps in nourishment. So this particular portion is going to be correct. Works. Perfect. Cool. Now, just a second. So this is marked wrong. Okay, this is going to be the correct answer. Some issue. Okay. And I have spoken about Pariksha Veera. If at all you still remember it, go and take it up. Yes. Now the last portion of, ideally not last, but last portion of this particular portion, that is male reproductive system, is pollen grain. As in, pollen grains, can they be acting as an allergen, or basically can they be acting as a performance booster? So listen to me. Yes, some of the pollen grains can cause allergies. For example, you might have seen this wild thing growing everywhere. This is known as parthenium. Okay, also known as carrot grass. Some people, whenever they get the pollen grains of this, they are like, chee. Okay, like that they go. Why? Because they have allergy. So yes, it can cause asthma, it can cause bronchitis, it can cause respiratory disorders as well. If at all in the option it comes, can it cause this? It does not cause any of this. You should know it causes asthma, bronchitis, as well as respiratory disorders. Example for the one that causes allergy is usually parthenium, that is known as carrot grass. Now you know pollen grain is there. It has vegetative cell that is there for nourishment of the generative cell. That means it has enough nutrients. It is highly nutrient rich, which is why people use it as food supplements as well. Some people in the Western countries, remember, okay, east or west, pollen grain is the best. So you have to remember Western countries. This was, I think, a neat question or something like that. Pollen products such as tablets, syrups are available, and they are very popular in Western countries. Not Eastern, it is Western. This question came. Which part? It is Western. Remember it. Okay, and as I said, it acts as a performance booster in the case of athletes and race horses. Why? Because obviously it has enough nutrients to enhance the stamina. Cool, ma'am. Done, ma'am. Done then. It's all cool. We just have one portion, and that is this. So listen to me very carefully. When I say that the pollen grain is there and it carries the male gamete, blah, 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 I mean that it's important for me, if at all, it lands on the female reproductive system and basically forms the pollen tube, remember, and carries the male gametes. This process is known as what? Pollen germination, where they actually form the pollen tube and carry the male gamete. Now, pollen pollen must land on this particular area, that is the stigma, that is the female part, before it loses its viability. Viability means what? Ability to probably go ahead and do the germination process and later on develop further for fertilization. Okay, what is viability? The ability to go ahead and do germination and later help with the fertilization process. So pollen should be landing before it loses the ability, right? Before it loses the viability is the correct term for it, right? So that is something that you have to remember. So in some cases, okay, in some cases the viability period is very less. For example, the things that we eat, okay, rice, wheat, everything. After every 30 minutes we are hungry after we eat it. Okay, so you can remember like that. They lose viability within 30 minutes, very less time. Okay, within 30 minutes we start feeling hungry also. But there are some pollen grains which have long-lived viability. That means they can remain viable for months. The pollen grain is released. After a few months they land somewhere, still they can germinate. Example is what? Rosaceae, Leguminosae, as well as you have Solanaceae. Okay, you have what? Rosaceae, Leguminosae and Solanaceae. RLS, Rosaceae, rose and all, leguminous plants, Leguminosae, and finally Solanaceae means basically your potato, chili come under Solanaceae family. These are the ones that have what? High pollen viability period out there. Got it? Got it. Next thing, pollen storage. So basically, since the pollen can anyway be stored, it has a good sporopollen content and a lot of that, things like that. And if I feel that there's one plant which is going to be extinct very, very soon, can I try to store the pollen grain of that and later do some artificial hybridization for crop improvement programs? I can. For that, I can have to store the pollen grains. How will I store it? I'm going to store it in liquid nitrogen. I'm going to store it in liquid nitrogen. For you guys who do not know, liquid nitrogen has a temperature of minus 196 degree Celsius. Liquid nitrogen, when I say that I'm storing something in liquid nitrogen, automatically the temperature is going to be minus 196 degree Celsius, which is pretty safe for everything. Okay, which is pretty safe for everything. It can be stored without damaging the cell, at all. So pollen storage can be taking place for what? For the crop improvement programs, for the crop breeding programs. Okay, and they can be stored in the seed banks as well, basically such that later on if at all we need, we can use them. But remember, liquid nitrogen and minus 196 degree Celsius. And with that, we have completed the androgen. Yes, we took 1 hour, correct, 1 hour to complete it, but I'm pretty sure any question that comes from microsporogenesis or probably the male gametophyte or probably this part, that is the pollen allergen or things like that, you will be able to answer it now. And now we're going to go ahead with the next part, that is about the female part, and we are going to make sure we do it a little quicker. And that's all. I'm just taking a break. This video is going to be stitched such that all of it appears to you together in one shot. So make sure you just go through your NCERT by this time. So we are back again with the chapter, children, and now we're going to go ahead with the female reproductive system. Just like how you had androgen in the case of males, right? Similarly, the female reproductive system is known as gynoecium. There you had a simpler name for androgen, it was known as stamen. Here also you have a simpler name for gynoecium, and it is known as pistil, or sometimes it is also known as carpel. So you have two names, it's the same thing only. Pistil, carpel means the gynoecium or the female reproductive system. Now this part of your chapter is going to be a little long, and probably there will be too much importance given to this, which is why I thought of explaining all of it within just one slide. Can you believe that? Yes. So I promise you, if you make sure to listen to me throughout this particular one slide out there, each and every concept related to the female reproductive system, and not just that, but also about fertilization is going to be clear to you. For fertilization, I might need one more slide, but still, it will be clear to you, so you have to listen to me carefully. Okay, let's go. So, Baba, the female reproductive system or your pistil or carpel is made up of few things. There is something like this. This part is a sticky part. Why sticky? Why sticky? Because the pollen grains which are gonna come, okay, they should be landing up somewhere. Suppose this portion is not sticky. What is going to happen? The pollen grains are la 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 and it went off. You do not want that to happen. You want it to stay there. Why? Because the pollen grains we studied, they have to germinate, they have to form the pollen tube, and then they have to form what? The Basically, they should be ideally sending their male gametes for the fusion with the female gametes, and that's how fertilization is going to happen, which is exactly why the stigma which is there, it has to be sticky in nature. Okay, that's the first part. Now the second part is the style. This long slender like thing is known as what? Style. This is where, so suppose your pollen grain landed up over here, it is going to form the pollen tube, and this pollen tube is going to move through this style, to reach where? To reach over here, which is known as, Baba, the ovary. It is known as the ovary. Ma'am, ovary is the female gamete? No. Ovary contains something else that has the female gamete. Okay, so female gamete is not somewhere near to us. No, listen to me. So this is your ovary, okay? And this is going to be the ovule. Ma'am, ovule is the female gamete? No. Ovule will be having something again that is going to have the female gamete. What is that something? Basically, just like, if at all you remember, okay, there was male gametophyte, that is pollen grain, right? Pollen grain had the gamete, male gametes. Similarly, over here, you have the female gametophyte. And what is the name of the female gametophyte? It is known as the embryo sac. Ma'am, this is the female gametophyte, not the gamete. No, it's not the gamete. Within this, the female gamete is going to be there. At the end, you will be finding the female gamete. So, ma'am, I have a question. In the ovule, the embryo sac is already there? No. We are going to discuss about how the embryo sac is going to be formed. Just like you remember, the pollen grains were formed after what? After microsporogenesis happened, the microspores were being formed. The microspores later developed into what? The male gametophyte, that is the pollen grain. Similarly, over here also, there's a process that is known as megasporogenesis that is going to take place. You will get the megaspore. From the megaspore, your female gametophyte, that is your embryo sac, is going to be made. And inside the embryo sac, you will be having the female gamete. But this is the outline. You have the ovary, you have the ovule. Within that, everything is going to happen. Okay, and I'll tell you one more very important thing. This ovule which is there, it has another name, and you need to remember that name. You need to remember that name. If you remember, inside the anther, in each corner, what did you have? Microsporangium. And the microsporangium did what? They formed the pollen grains later on. Similarly, over here, ovule is known as megasporangium, because this is where the female gamete, or basically the female gametophyte, is going to be made. What do you call it? Megasporangium. Okay, you call this as megasporangium. Okay, done. Right? So this is megasporangium. Inside it, everything is going to happen, just like how you had in microsporangium out there. Right. Now, if at all you have been asked about, draw a diagram of megasporangium and explain. Just understand, you have been asked about the diagram of ovule. So, ma'am, we'll just make it like this, one circle, and then it's done. Ovule is done. Right? No. For that, you have a detailed picture given in your NCERT, which could be complicated at times, which is why I am going to make a simpler image. So, listen to me. This is going to be your, just a second, yeah, this is going to be the ovary. Done. Okay, ma'am, clear, ma'am. This thing that is there is going to be the ovule. Now I'll show you magic. When I click on this, it will come. Yeah. Okay, this is going to be what? Your ovule. What's the other name for ovule? It is known as megasporangium, ma'am. Okay, let's make it megasporangium. Cool. Done, ma'am, done. Then what? Do you think it's just staying in the air like this? Obviously, it cannot stay in the air like this. You need a support. Who provides the support? So listen to me very carefully. This ovary which is there, it will be having a cavity over here. So much space. Okay, this is known as the ovarian cavity. This is known as the ovarian cavity. And if at all you had to give it a term, you would actually call this as locule. Locule is nothing but the cavity which is there, the ovarian cavity which is there, you have given a specific name, which is why you call this as locule. And obviously, it does not stand like this in the air. You need to support the placenta. Sorry, you need to support the ovule. For that, you have the placenta, just like how we have placenta in our body, right? Similarly, for nourishment purpose, they also would be having a thing that is known as placenta. And it is super important to nourish this guy. Why? Because this is where everything is going to happen. So we have a placenta, and obviously placenta and this has to be connected. For that, we have one next thing out there, which you call as, come on, work. Okay. Yeah, it is known as funicle. It is known as funicle. And the funicle goes and says, hi ovule macha, how are you doing? So which is why this point of attachment which is there is known as hilum. Okay, so it's just saying hi, you can remember like that. So this point of attachment is known as hilum. You have the funicle which connects the placenta to the ovule. Done. Now the ovule says that, ovary, I feel very cold over here. Can you give me sweater? So then the ovary says, yes, please do. Obviously, make sure, consider this as your home. So over here, you have a sweater. This sweater is known as integument. Integument is actually there for protection. What is it there for? Protection. So you have an inner integument. You have an inner integument. Similarly, outside this, you have an outer integument. This is inner integument and this is outer integument. Ma'am, why you are not closing up? Arey, because it's a Gen Z, not just Gen Z, even millennials like me, all of us basically do not like putting up the sweater like a full zip up. We are like a little style here and there. We probably knock it down somewhere open, right? For extra air to come in. So just similar to that, over here, Baba, okay, this guy is also not ready to wear the sweater fully. So there is a small space over here, and this small space which is there is known as what? It is known as the micropyle. It's a pore actually. It's known as a micropyle. What is it known as? Micropyle. And this area where the micropyle is there, I'll just call this as micropylar region. Micropylar region. Now, there's one very important thing that you need to remember. Wherever the micropyle is there, you call this as micropylar region. Agreed, ma'am. No issues with that. Do you have a separate name for the region that does not have the micropyle? The answer is yes. The opposite end of wherever the micropyle is, you will call this region as the chalazal end. What do you call this as? The chalazal end. So we have micropylar end and we have the chalazal end, and this is how generally a megasporangium is going to look like. So ma'am, you actually spoke about everything else that is there in ovary, but not actually exactly what's there inside the megasporangium, because that's a diagram all about. So listen to me carefully. Inside the megasporangium, that is your ovule, you will be having so many cells coming together. So many cells over here are going to come together. This mass of cells which are there, okay, you have a name for it. This mass of cells is known as nucellus. It is known as nucellus. Okay, just like if you remember microsporangium, inside that you have sporogenous tissue which was made up of so many cells, but there all the cells were able to form the microspores out there later on. That is why they were all known as pollen mother cell or microspore mother cell. Here it's not like that. There are some cells over here for nourishment, and all these mass of cells are known as nucellus, but apart from it you have only one cell over here. And this cell is known as megaspore mother cell, which is going to undergo megasporogenesis. Only one. There everyone was dividing, but here only one person is there, and that is known as megasporogenesis. So let's put a queen's, crown over here. So you have the megaspore mother cell. Inside what? Inside the megasporangium. Apart from it, for the nourishment purpose, you have mass of cells, which is now known as nucellus. Cool. This is what you need to know. Now, to make sure that everything has settled down in our brain, what do? We just read it. So what do you have? Placenta. Placenta is located inside the ovarian cavity, is it? Yes, Mama. This is your cavity, and over here you have the placenta, so this is inside. Okay, ma'am. Funicle, stalk attaching ovule to the placenta. Funicle, this is a stalk attaching ovule to the placenta. Done, ma'am. Okay, cool. Sorry. Now, hilum, junction between funicle and the ovule body. Hilum, junction between ovule and the funicle body. Done, ma'am. Done. Okay, ma'am. Next thing, integuments, 1 or 2 protective layers leaving a small opening that is known as micropyle. Okay, 1 or 2 protective layers leaving a small opening that is known as micropyle. Done, ma'am. Then micropyle, small opening at the tip of the pollen for the pollen entry. We didn't discuss that. So listen to me carefully. Why do you think there's an opening over here? It is because the pollen tube which comes, right, it has to actually enter the ovule, because inside the ovule you have the embryo sac later on, and then then the female gamete, right? So for that you need an entry point, and that entry point is actually the micropyle. It is going to be the micropyle. Always remember that. So micropyle is there as of now for the pollen entry. Later on, when fertilization happens, its function also changes. We'll talk about that later on only. Okay, you remember that yes, ma'am has to talk about what happens after fertilization. Anyways, then we have chalaza. The opposite end of the micropyle is the base of the ovule. Yes, the opposite end of micropyle is known as chalaza, and that's the base of the ovule. Done, ma'am. Then we have nucellus, mass of cells with reserved food. Yes, I already told you this is a cell that has reserved food, and this is there for nourishment purpose. Done. And then we have a mighty megaspore mother cell. This is a thing that you need to know about megasporangium. If at all you have been asked to draw a diagram of it, this is the diagram that you have to draw. In your NCERT, it could be twisted, turned and all. Do not be worried. This diagram would be more than enough, and the teacher is going to give you marks. Make sure you label all of it in one side and in the straight lines as well. Cool. And the diagram is important. You need to study it. Now, since I'm already talking about this, I want to tell you one very important thing. And you have to listen to me very carefully. Sometimes the ovule says that I'll get inverted. I am going to get inverted. I do not like to stay like this. I want to get inverted. So what exactly happens is that this placenta and the funicle and the point of attachment, that is hilum, it is getting shifted from here to here towards the micropyle. Towards the micropyle, it is going to get shifted. So suppose this is shifted, suppose this thing is not there. Will you still call this end as chalazal end? Yes, because that's the opposite side of micropyle. That's the only thing that you have to look forward to. This can come over here. In that particular case, when you have an inverted ovule, please remember the term. I'm going to tell you a term. Inverted ovule which is there, and where should I write it? Here. Okay, so inverted ovule is known as what? It is known as anatropous ovule. Anatropous ovule. And is the diagram of anatropous ovule important? Yes. How do you draw it? Nothing. You just put this part over there. Make a placenta over here. This is your funicle, and here is your hilum. Done. So easy. That's the only thing, and you have to remove it from here. Chalazal end, everything remains the same. Anatropous ovule is what you call it. The diagram have been asked, and this is what is given in your NCERT. So over here, if at all you look at it very carefully, you can see this is the pore. This is the easiest one to identify. This is your micropyle. Okay, these two would be your integuments, yes, and this would be your ovule which has nucellus, and later on, as I said, it will be forming one megaspore mother cell which is there. It is going to later on form the megaspore, and that megaspore forms the female gametophyte which is known as embryo sac. The opposite end of micropyle is known as chalazal pole. Done. Okay, over here, if at all you see, there is a structure like this, big structure like this. This is your funicle. I have mentioned it as funicle, and basically it is the point of attachment that they have is known as hilum. It is known as hilum. Obviously, the top of it you have the placenta that is not made over here. So to give them also an easier picture, you can draw something like this. This works. Okay. So this is the structure that you need to know. This is everything that you mostly need to know about the female reproductive system as well as its structure. Now, there are a few other things also which I need to say, but I'll keep them on hold for now, and I'm going to go ahead with this scene, because anyway we are over here. We want to see what is happening, what drama is happening over here with the megaspore mother cell, who is the only one, in the kingdom out there. So the megaspore mother cell says that, Macha, you know what, I'm the queen over here. I decide what happens. I am going to undergo megasporogenesis. So the next process that we're going to learn about right now is going to be what? Megasporogenesis. How the megaspore mother cell is going to form the megaspore. Is it exactly like the microsporogenesis? All of it is going to be discussed over here. So listen to me very carefully. Here you have the megaspore mother cell, right? What is a ploidy? What is the number of chromosomes up there? Ma'am, a normal cell, just like a normal cell, it will be having 2n, because I remember that you said that when we start with diploid cell, and after first round of meiosis, we will be having half the number of chromosome, and you will now call this as haploid. What happens in meiosis 2 then? The number of ploidy, it remains the same only, ma'am. It remains haploid only. Okay, Macha, all cool. So here also is the same thing. You are going to get 4 megaspore. You are going to get 4 megaspore. But now comes the twist, and this is the saddest part in the female's life. So what exactly happens is that it formed the 4 megaspores, but you know what? 3 of them are going to get degenerated. Tata, bye-bye. So ma'am, why did we do all of that? It just had one megaspore mother cell, and basically we killed 3 cells, megaspores that it formed, and we just have one. What are we going to do with the one? Well, listen to me very carefully. This one which is there, okay, takes care of it really, really well, and this megaspore forms the female gametophyte. Only this one. Only this one is going to form what? Ayyo, black color. It is going to form, come on, work, yeah, the female gametophyte. And what is a female gametophyte? Ma'am, we all know by now, it is known as what? Embryo sac. So let's write it. It is known as embryo sac. This is your what? Your female gametophyte, which is known as embryo sac. And this is the only one who does it. So do we have a name for it? Chutki, Butki, Chitku, Chutku, Chunnu, Munnu, anything like that? No, you have actual name for it. It's known as functional megaspore. This is the only one that function well. Is it because of that you say that? No. So functional megaspore is the only one that goes ahead and probably gives rise to the female gametophyte, which is why you call it functional. Now the next thing is that this type of development where only one megaspore is developing into the female gametophyte, not all of them, you call this as this type of development where only one goes ahead and forms the entire embryo sac is known as monosporic development. It is known as monosporic development. Remember this. So you have the female gametophyte, that is the embryo sac, and it is formed by just one megaspore. It's the only one that develops further. That is why you call this as monosporic development. So if your question comes, why it is known as monosporic development? Because only one functional megaspore is there. That's why. No, because only one functional megaspore later on develops into the female gametophyte. What does it develop into? That is more important. Got it? That's all. Okay, now listen to me carefully. All of it is happening where? Inside the ovule that we just studied, right? We studied over here about the ovule. This is where everything is happening. This is where the division happens, blah, 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 blah. Everything happens. Where does the division start? You know, where does microsporogenesis start? It happens here in the micropylar region. It starts in the micropylar region. Always understand that. Okay. Now you got 4 cells, ma'am. Out of them 3 degenerated and only 1 remained functional. So is there a location difference where these 3 of them, they go somewhere and that's how they get degenerated or something like that? Yes. So basically 4 of them are formed. Okay. In that Chutki, Butki and Utki says that, Macha, let's go to micropyle. Okay. Anyway, we have divided, we have come far over here. Let's go back to the micropyle and have some chill time. Let's not include Puchku there. Okay, so Puchku is the only one who stays back. Rest everyone goes there to have ice cream. And you know what happens? They die. They get degenerated. Where? In the micropylar region. So listen to me very carefully. This starts in the micropylar region, megasporogenesis. Done, ma'am. Okay. Now we got 4 cells. Out of them 3 go to eat ice cream. Okay. And they get degenerated there. Only one that remains over here in the chalazal end. That remains functional. So this is the moral of the story. I'll tell you, do not go without your friend. You should be going with all your friends to have ice cream. The point is, my dear children, I told you in the story format, but the thing is, remember, usually everything good that happens in the micropylar region. You saw that megasporogenesis started, and yeah, that is the only good thing that have happened so far, but more good things are yet to come. Okay, I'll talk about it. And usually the bad things happen in the chalazal area. But this time, this is the only case where good thing happened in chalazal area. The one megaspore that is there remains functional in the chalazal area. And the 3 of them that get degenerated happens in the micropyle. This is the only exception that we have. Remember that this is a KCET question that has come. So, yes. Now we come over here. Where are we? Yes. So we spoke about this, and now I'm going to read about this. Megasporogenesis, formation of megaspore from the megaspore mother cell. Now MMC is microspore mother cell, MMC is megaspore mother cell. Do not get confused. It's easy because you know sperm, chotu, micro there, right? So here it's mega. Cool. MMC location found in the micropylar region. I told you MMC location is where? At the top in the micropylar region. Okay. MMC division undergoes meiosis producing 4 megaspores. Does it? Yes. 4 megaspore. Only 1 megaspore remains functional. The 3 degenerate. Okay. Monosporic development. Single functional megaspore forms the embryo sac. That is the female gametophyte. And now we're going to talk about that. How does this single guy over here does the entire job? does the entire job. By the way, I had made the nucleus over here, so you have a cell also. Okay, don't worry. This is a cell, this is a cell, the cell divides, the cell divides, the cell divides, the cell divides, the cell divides. Inside the cell, you have a nucleus which is haploid in nature. Okay. Now we are going to go ahead and see how does the transition happen from what to what? From your megaspore. Children, by the way, I hope you are understanding things. If you are, let me know in the comment section that ma'am, nice lecture. If not, ma'am, boring lecture. Okay, so I'll be deeply hurt if at all you say that, but it's okay. I can improve. I'm here for that. Okay, so we have the functional megaspore. Okay, and that transforms into the embryo sac. That is a logic. That is something that we are gonna study right now. So listen to me carefully. I'll just increase the brightness and calm because I think that is needed now. So, okay, perfect. So over here, this is, for this I do not have to make the diagram. The diagram will already come up. Just a second, yeah. So this is your embryo sac out there. Yeah, sorry, this is your what? Functional megaspore out there. This is your, where is it? Come on, work. Yeah, this is your functional megaspore, and we are going to transform into what? Embryo sac. Embryo sac will come over here later on. So this has a nucleus, okay, and the nucleus is what? It's haploid in nature. You know what happens? The nucleus says that I am going to start with division, and I'll make sure that when I divide, I will not be making a cell wall around me. I will not allow the formation of cell wall around me. First of all, I wish to divide myself. The nucleus just want to divide itself and increase the number. So what division is going to happen? Where they just want to increase the number and the number of chromosomes remains the same, it is going to be mitosis, not meiosis. In meiosis, if at all you do, it becomes half. So you want to maintain the constant number of chromosomes, you just want to increase the number of cell, or basically the nucleus are there. It is known as mitosis. So over here, the nucleus divides into two. The number of chromosomes remains the same. Yes, ma'am. Obviously, it is going to remain what? Haploid only. It is going to remain haploid. Come on, what's the problem? Okay. And this is what? This is the end result of first mitosis. Okay. Today, my patience is being tested. First mitosis. So, ma'am, you are saying nucleus has been divided, cell wall formation is not taking place. Yes, cell wall formation is not taking place. Okay, ma'am, again it divides? Obviously, yes. So, again over here, you have the second division, second mitosis. Again mitosis, ma'am, again mitosis only. Okay, so that means that this cell which was there, this nucleus which was there, it divides into another two nucleus having haploid number of chromosome. Ploidy remains the same. Why? Because it is mitosis. So now we have got 4 nucleus, ma'am. Yes, 4 nucleus. Okay. Now, again does it divide? Oh my God, it does not end? It ends. Listen to me very carefully. So over here, you have what? You have the third mitosis. This is going to be the third mitosis, and that's all. That's the end of the story. That's the end of the story. Okay, ma'am, that's the end of the story you are saying, but I don't trust you. Let me just hear what you're going to say next. So this N divides into 2 nucleus having NN. So we have two nucleus. That means later on it will form 2 cells. Again NN, 4 cells now, 2 plus 2, 4. 6 cells and 7 cells. It becomes cells when you have cell wall around it. As of now, it's not like that. As of now, it's just mitosis, and please do remember, 3 mitosis happens. This is again a question. 3 mitosis happens, and all 3 of them is free nuclear division. Is going to be free nuclear division. What do you mean by that, ma'am? Free nuclear division means the nucleus is dividing constantly, it does not have cell wall formation happening. Okay, the nucleus is kind of free. That is why you call it as free nuclear division. Okay, ma'am. At the end of it, it will make cell. No. Yes, but there's a twist, and I'm going to show you the twist. So over here, okay, over here I have the embryo sac. Now I have got the nucleus and all. Now I am going to arrange them to form the embryo sac. From functional megaspore, now we're going to form the embryo sac. 3 of these cells go at the top. Okay, 3 of the cells. I mean, cell wall is formed around them, around the nucleus, and 3 of them arranged at the top. What do you call this as? Antipodal cells. Ma'am, the ploidy is going to be haploid, haploid, haploid. No, obviously, yes, haploid, haploid, haploid. 3 cells done. So out of this 3 done, so I'll just cross it off. Cross, cross, cross. Okay, cross, cross, cross. Okay. Now we have 1, 2, 3, 4, 5. 5 of them left. Okay. Now 2 of them come over here. They are known as synergids. They are known as synergids. Again, they also have nucleus. The nucleus is going to remain haploid only, ma'am. Yes, obviously that remain haploid. So again, 2 cells gone. 2 cells. Now we have 3 cells left. Okay, one more cell joins up over here and it is known as the egg cell. Oh my god, everyone do like this. This egg cell is what? The female gamete, the one that we have been waiting for so long. This is going to be what? The female gamete. Okay, ma'am, got it, ma'am. This is also definitely going to be haploid. Obviously, yes. Okay, now you just need 2 cells and then it's done, right? So we have got 6 cells, 3 here, and everything is sorted. So let's make the next cell. Okay, there's no cell. Is it done? Is it done? Yes, it's done. Listen to me. Here comes the twist. And here you see the 2 nucleus are joined together. I mean, basically they are together. And I have given a name for it. 2 polar nuclei. What is this thing? Where is our cell, ma'am? So listen to me carefully. This whole region that you see over here is a very big cell. And you know the name for it? Okay. The name for this particular very big cell is known as central cell. This cell which is there, it is a 7th cell. It is a 7th cell and it says that since I'm a very big cell, I really need 2 nucleus. I cannot have 1, I need 2 nucleus. So there is a problem. How many nucleus did we had? 1, 2, 3, 4, 5, 6, 7, 8. We had 8 nucleus. We had 8 nucleus. And now how many cells do we have? We have only 7 cells because one of them decided to basically have 2 nucleuses, to keep 2 nucleus. So remember always, and now by the way, we have formed the embryo sac. An embryo sac has a condition which you call like this. It is 7-celled, 8-nucleate in nature. Or basically 8-nucleate, 7-celled condition is there. Always remember, one very big cell is there that needs 2 nucleus. That is why 1 nucleus which is there, it becomes extra. The cell is going to be 7, nucleus is going to be extra. They can give you 7 nucleate, 8 cell condition. Don't fall for it. Read it carefully. 7 cell is there, but the 7th cell says that I need 2 nucleus. And over here, the antipodal cells are at the top. This is at the top, by that I mean chalazal region. This is basically over here in this picture, it is top, actually it is in the chalazal area. Okay, where are these things? Synergids as well as the egg cell. This is going to be there in the micropylar region. And let's use a tiny little brain over here. Macha. Over here, we wanted the pollen tube to come in, and pollen tube carries what? The male gamete. The male gamete would love to meet the female gamete first, no? Which is exactly why you remember micropyle is over here. It's over here. The opening is over here. It wants to meet the male gamete very, very soon out there. And the synergid and the egg cell together. The synergids are actually best friends with the egg cell. You call all of them together as what? The egg apparatus. You call it as the egg apparatus. This is very, very, very important. What do you call it? Egg apparatus. Cool? Fine. Works? Okay. And also remember one more very important thing. Over here in the synergids, you have some finger-like projections like this. You have, where is it? Come on, darling book. Okay, so over here you have some finger-like projections like this. What is the use for this? Basically, when the pollen tube comes, it's like, sir, please come in, welcome. Go through this particular synergid, you will reach your female gamete. So just to guide the pollen tube, what do you have? You have your filiform apparatus. This thing is known as filiform apparatus. Okay, it's known as filiform apparatus. It's the finger-like projection that you have, which is there for guiding the pollen tube. This is important. Guiding the pollen tube. Again, an MCQ question. Only guiding the pollen tube. Got it? And that's all. And that's how we have made the embryo sac. So my dear children, our journey have been long for 30 minutes. What did we discuss? We discussed about how exactly the female, reproductive system looks, stigma style. Ovary is there. Inside the ovary, we saw the structure. Inside the ovary, there was one cell. We decided to follow the story of that one cell. That one cell divided into 4 cells. 3 of them degenerated. 1 of them left back. And that one now has become what? An embryo sac, which has 7 cells and 8 nucleus. This is very important. You will be asked to draw the diagram of an embryo sac. You have to draw it like this. Label everything neatly and you will get your marks. Okay? Got it? Yes. Now I can go further and probably talk about double fertilization here itself, but I'll restrain myself from doing that right now because we are in some other topic, that is a female reproductive system. So we'll complete it. Okay, then we are going to see about pollination, as in basically how does we form the pollen grains, we form the female gametophyte. That means we have the male gamete, we have the female gamete. We will see pollination first and then we will see fertilization. That is how the chapter is going to go. Okay, so until now, I hope each and every part of the story is super clear to all of you. Let's have a quick read. Just a second. This is all animation that I put up. Okay. Let's have a quick read, okay? So, functional megaspore nucleus divides, okay, becomes 2 nucleate, becomes 2. Further mitotic division, 4 nucleus, and then 8 nucleus at the end. No immediate cell wall formation happens. This is known as free nuclear division. No immediate cell wall formation happens, and please remember, it's 3 mitosis that take place. 3 of them takes place over here. In the case of male gametophyte formation to form the male gamete, until then you required 2 mitosis. One was to form the male gametophyte with the 2 cells. Okay, microspore divided into 2 cells, 1 mitosis. And then the generative again divided to form the gamete, so second mitosis. But here it is 3 mitosis that is there. And there it was followed by cell division, but here it's not like that, it's free nuclear division. Now, after 8 nucleate stage, cell walls form organizing the embryo sac. Cell distribution in the embryo sac, egg apparatus. What is egg apparatus? In the micropylar end, you have the egg cell plus 2 synergids with filiform apparatus for pollen tube guidance. You know what? This papa guy that is a synergids, I told you they are best friends. You know, it dies for this particular egg cell. When pollen tube forms, no one of them is going to degenerate. That's the friendship all about. But don't die for your friend right now. Please live for them. Okay. Now, central cell that we have contains 2 polar nuclei. Agreed, ma'am. That selfish cell which was having 2 nuclei. Okay. And antipodals which is there contains 3 cells out there. And they are at the chalazial end. I told you, everything that is not useful happens in the chalazial end. Everything that is useful happens in the micropylar. The only exception was where the 3 degenerated in the micropylar and 1 that remained functional was in the chalazial. Okay. Mature embryo sac, 8 nucleate but 7-celled structure. Done, ma'am. Done so. Now over here, listen to me. I have to tell one more very important thing. Oh my God. Okay. So, when I talk about all of them, you see all of them are going to be haploid in nature, right? The cells are going to be haploid, having half the number of chromosomes, but there's an exception. This central cell which is there, it has 2 nuclei. N and N becomes what? 2N. It is the only one which is going to be diploid. It is going to be diploid in nature. Everyone else in this embryo sac is going to be haploid except the central cell which is going to be diploid. Why? Because it has 2 nuclei. This is about the ploidy. This is also something that you have to remember. Okay. So we have studied almost everything about it, and now I come back to this. So we started off actually with this page, that is gynoecium, where we spoke about pistil. What is a pistil made up of? Stigma, style and ovary. Stigma is the landing platform, style is a slender stalk below the stigma, and then ovary is the basal swollen part. Ovarian cavity is there, that is known as locule, which in turn contains the ovule, that is known as megasporangium. Yes, we have the megasporangium within it. Okay. Now over here, the next thing that we have not touched until now is the types of pistil. So there can be a flower which has multiple pistils out there. Stigma style ovary, stigma style ovary, stigma style ovary, multiples of them. Okay. And there are stigma and basically there are flowers which have only one. That condition where it just has one, you call this as monocarpillary condition, not monopistillate. It's known as monocarpillary condition. Monocarpillary means mono means one. Only one carpel is going to be stigma style ovary. There are conditions where multiple carpels could be there. That means multiple or female reproductive parts could be there in the same flower. Okay, and in that you have 2 situations. One is where, just imagine, okay, one is where stigma style is there, so many stigma style is there, but ovary of all of them is fused together. Ovary of all of them is going to be fused together. This condition is multicarpillary, definitely multiple carpels are there, but this condition where all of them are fusing together is known as syncarpous condition. You can remember synchronization, okay, just like that, they are in sync. Okay, this is known as syncarpous condition. Cool. And there are situations where all of them are separate. That means there are so many carpels, but everyone has their own ovary. Basically, they do not have to rely on anything else. That is known as carpus condition. Now in your NCERT they have given you examples of syncarpus as well as apocarpus. The picture that they have given is something like this. Now with this particular diagram, very beautiful diagram, we do not understand anything, right? What do you understand? I do not understand anything. Syncarpus ovary they have mentioned, okay, and this stigma is what they are showing over here. This is stigma and this is ovary, okay. What do you mean by that? Basically it means you will have multiple stigma, you will have multiple style also, but the ovary of all of them is going to be fused. This picture is actually of this particular plant. Remove all this thing. This is basically the pollen, the anther and the filament. Okay, this portion is one that is being made over here. Okay, and this thing that you see over here is your stigma. Stigma will be having style, but all of them are fused over here within having only one ovary. Ma'am, where do you see this? What is this flower? I do not know. You know, this is a dangerous plant. This is papaware. This is what? Papaware. You are going to study this in human health and diseases. It's a plant that produces a drug that is opium or heroin. Okay, so basically, let's go ahead and remember the name. It's known as papaware. Okay, when I was not able to remember it, I remembered it as Tupperware. You know, Tupperware bottles were there. So anyway, do not worry about that. This is papaware. Okay, this is papaware. Cool. And then you have apocarpus condition where I told you everyone is separate. Again, they have given a picture in your NCERT that looks like this. Now, what do you understand from this picture? I'm so sorry to say, CBSE board, you should definitely make sure children understand with the help of some good pictures are there. So this is the carpel that they have mentioned. You know exactly how it looks? It looks like this. You know where is this particular thing? In this particular flower. This flower smells so good. This is known as champak. In Malayalam we say chembagam. By the way, any Malayalis up there? Okay, anyways. This is known as chembagam. Okay, and Hindi it's known as champak. It smells so good, right? Yes, so within this particular flower, if at all we get to see something like this. They have stigmas, these all things that you see is stigma style ovary. Not ovary, you cannot see it, but anyway, they have all of them as separate thing. This is not unified together. They're all separate. Okay, now you cannot write champak and chembagam and things like that, which is why you have an actual name to write, which is known as Michelia. This is a scientific name. It is known as Michelia. So remember, papaware is there where you have syncarpus condition, and so all the papas in the world are in sync. That you should not be doing anything apart from studying from this time to this time. After that, you can use your mobile. So they are always in sync. So syncarpus, papaware. Okay, and this is Michelia, who basically has is very beautiful over here and has all the female reproductive part in separate positions. Okay, they are not fused. So pistil we studied monocarpellary condition and multicarpellary condition. Okay, and multicarpellary, in that you have two examples which you need to remember. So yes, we have discussed this. Now we come to this. So now listen to me very, very carefully. Okay, I'll tell you a very, very important thing. This ovary that you see over here, this is the ovule, this is the ovary. Inside the ovule, what is happening? Embryo sac is being formed. Now you have the female gamete. What happens if fertilization is there? If one fertilization is there, basically the male and the female gametes fuse together over here within the ovule. And at this point where the fusion has taken place, fertilization has taken place, I am now going to say that basically zygote is being formed. I will say zygote is being formed. Or then later on the embryo is going to be formed. Okay, that is a basic logic out there. At this point, when fertilization happens, always remember the ovule becomes a seed. It becomes a seed inside which the embryo is there. So that is why whenever you have a seed and you put it somewhere in the soil, it grows up. Why? Because the embryo is there. If at all you give them nourishment and everything, it is going to grow into a new plant. So ovule transforms into what, Baba? Always remember, after fertilization, post-fertilization, the ovule transforms into a seed. What about the ovary? The ovary transforms into a fruit. It transforms into a fruit and ovule transforms into a seed. This is the basic logic that you should know. Now, in your NCERT, they have given you some condition. Okay, they have given you some examples. There are some plants with multiple ovules. How do you remember it? With using our brain. Listen to me very carefully. Where do you see multiple ovule? Multiple ovule means multiple seed. In which plant do you have multiple seed? Watermelon, ma'am, papaya, ma'am, all of them have multiple seed. That means they have multiple ovules also. These ovules are the one that forms the seed. No. So remember, the easy ones to remember for you, because you know that you have eaten that, it will be papaya. Okay, it will be papaya, and the other one will be watermelon. But there is one thing that you might forget, and that is orchid. Please remember this. Orchid is the next thing that you have, and you need to remember this. It's a very, very, very important thing. Cool, ma'am. Okay, now over here, there are situations where only single ovule is there. Only one ovule. That means only one seed. And that is there in the case of cereals that we have. Wheat, paddy, all of it. Similarly, mango is there. Mango, if you have seen, it just has one seed. Have you seen multiple seeds out there in mango? No. That means one ovule. It's going to be single ovule. Cool? This is very, very important. Please not forget it. Cool? You can get questions based on it. So this part of the story is also clear. Now, what questions have come from this part? Whatever you have studied, as I told you, you are asked to draw a neatly labeled diagram of a anatropous ovule. You have to draw it. Funicle and hilum would be on this side. That's the only thing which is there. If this picture helps you, you can look out for this picture. This is 3 marker. This came in MQP, that is Model Question Paper 3, the previous year. Now, the next question that we have, oh my God, yeah. I've shown you the answer, but still we can try. Given below is a diagrammatic sketch. This is a KCET question. Of anatropous ovule, select this correct set of names of parts labeled as A, B, C, D. So let's see. Over here, we have A, this is B, this is D, and this is C. I'll tell you the easiest to find out will be what? If this is the micropyle, the C is going to be chalazial end. Let's see. C is chalazial. I have 3 portions like that, 3 answers like that. So I'll cut this at least. This is the odd one out. We do not need it. Okay, cool. Now we have to spot the next one. So C we know is chalaza. Okay. Now the next thing that we should be knowing is that over here, this particular portion which is there, they might be either indicating hilum, that is the point of attachment, or they could be indicating funicle. So let's see, A can be hilum or funicle. So funicle is there and nucleus is there. So obviously this portion also goes off, because A and C is fixed over here. Funicle, chalaza. Now from this option we have to find out. D is given as micropyle over here. Micropyle, there was no tag that was given. D micropyla. Micropyle should be here, no? So this is wrong. This is something else. Even if you do not know, you know that D is definitely not micropyle, which is why you can cancel this. What are you left with? You are left with C option. Listen to me very carefully. You should know that over here, this is your ovule. Inside the ovule, the outer layer is nucellus, and the inner one would be the female gametophyte, that is the embryo sac. So D is going to be embryo sac and B is going to be the nucellus. Okay, that's how we gonna solve questions. And then we spoke about megasporogenesis. We spoke about everything which was there. Yes. Okay. And here is a question. Again, this is a KCET question. This is an important question, a different question, which is why I kept it. Okay, so if a tetraploid plant contains 48 chromosomes in its nucellus, then the number of chromosomes in the egg and in the synergid respectively are. First of all, listen to me. Imagine the plant. There is a plant over here. Okay, there is a plant over here, and you have been asked, it's a tetraploid plant. What do you mean by tetraploid? Tetra means 4 and ploidy means n. Basically talking about number of chromosomes. Okay. Now it says that 48 number of chromosomes is there in the nucellus. Nucellus is what? A normal cell that is there for nourishment. It is going to be having the normal number of chromosome only. So if a plant itself is foreign in nature, obviously the nucellus is also going to be foreign in nature, and by that foreign I mean it is having 48 number of chromosome. So usually, basically nucellus is going to be diploid, because the plant usually are diploid. But here you are talking about a tetraploid plant, and in that case they have mentioned that nucellus, the cells of the nucellus is 48 number of chromosome, which means that here 4n is equals to 48. Okay, done. Then the number of chromosome in the egg and the synergid is, you have seen that every part of the embryo sac except the central cell, everything is going to be having half the number of chromosome. If a plant has this much number of chromosome, that is normal number of chromosome, the egg cell, the synergids and the antipodals will definitely have half of it. So you do not have to think that, okay, I have to make 4n as n. You should not be thinking that I have to make 4n as n. You just think I have to make it half. So here if it was 48, half of it is going to be what? 24. So in the egg cell as well as the synergid, it is going to be 24. This is just given to confuse you guys. Always remember, they can give you this number of cells is there in the nucellus, or this number of chromosome is there in the nucellus. Its nucellus is a normal cell. If this has this much, then the egg and the synergid will have half of it. Okay, they can give you another question. If basically the there is a root and the cell of the root has 48 number of chromosome, what is the number of chromosome in the egg and the synergid? A root is a normal cell, it will be diploid, it is basically going to have total number of chromosome. Any which way you do, egg and the synergid will have half of it. So even if you make tetraploid, polyploid, pentaploid, octaploid, anything, you just have to look, are you talking about a normal cell? If you're talking about normal cell, make it half. If you're given the number of eggs and chromosome in the egg and the synergids, and you want to find what is there in the normal cell, you make it double. That is how it goes. So the answer for this question is going to be what? 24 and 24. If you're not able to understand it, just go back, listen to it properly. I bet you will understand. Okay? Normal cells, half of the chromosome we have to take into consideration when we talk about egg and the synergid. Okay. Now the question is, what is the number of chromosome in the central cell? Central cell is what? We know it's made up of two nuclei. N and N comes together over here, right? So if we started with 48 number of chromosome and we said that haploid N is basically, or half of it is 24. So here this is 24, this is also 24, 24 plus 24, it becomes 48. Central cell was always an exception. It will have the number of chromosomes same like a normal cell. Why? Because it has two nuclei. Okay, so this is how the questions could come. Please be aware of it. And now we move ahead with the next topic on the 47th minute, and just give me a second, let me drink some water. I know this one shot is going to be lengthy actually, and you're seeing that, and I'm pretty sure everyone who's probably seeing that is getting the confidence that yes, this chapter is getting easy for me and I know everything in this chapter that could be coming for my examination out there. I'm showing you questions for your PU2, I'm showing you questions for your KCET. So yes, do not be worried, even if it's time taking, finish it off. Okay? And please drink some water. Very important. Now, you're gonna go with the next thing, that is type of pollination. So, Baba, in pollination also, I would like to tell with the help of a picture, because I think pictures help better. Drawing helps better, right? Pollination is what? Transfer of pollen grains. Transfer of pollen grains from the anther to the stigma. That is pollination. Pollination means fertilization? No. Fertilization means fusion of gametes. So first of all, pollination has to happen. Okay, pollen grain should land. Then the pollen grain will be interacting over there and forming the pollen tube. Now pollen tube is coming and basically should be transferring the male gametes. If the male gamete and the female gamete fuse together, that is known as fertilization. Do you think that every pollination that happens results in fertilization? Can you guarantee that? No, ma'am. They might go there, but they might not make the pollen tube, or probably there could be multiple pollen tubes being made, only one could reach there. So we cannot guarantee, we cannot say that pollination always results in fertilization. We cannot say that, remember that always. Okay, now one question. Fertilization, for it to happen, do you need pollination? Yes. Obviously, for fertilization to happen, pollination has to happen, then only the pollen tube is going to be formed. No. So for fertilization, yes, pollination is needed. But does pollination end up in fertilization for sure? The answer is no. Okay, that is one logic that you need to remember. Questions have come. Okay, now listen to me. There is a flower like this. And this flower is going to be an open flower. By open flower, I mean the male and the female part is just exposed like this. Then there is another flower. Okay. Then there is another plant. Okay. Now listen to me very carefully. First situation is within the same flower. What is happening within the same flower over here? Pollen grains are gonna land up on the stigma of this particular flower. Same flower, this process is known as self-pollination. Self-pollination, or you broadly call this as autogamy. Autogamy. Now, if you want this to happen, there are some conditions that you have to follow. The flower has to be bisexual. By that I mean it should be having the male part as well as the female part. Otherwise, how does self-pollination happen? So the flower has to be what? Bisexual. Next part of the story. Apart from the flower being bisexual, what is the next thing that you need? They have to be close to each other. The anther and the pollen has to be close to each other. Otherwise, how do pollination will happen? So the anther, or basically the male and the female part, you can just write like that. The stamen and the pistil. Stamen and pistil should be close. Okay, should be close. Okay, done. Next thing that you need is that not just being close, but you should understand the pollen should release the pollen grains the anther should be releasing the pollen grains at the same time where the stigma is okay to receive it. So there's a term that is known as receptivity. Okay, stigma should be mature enough to receive the pollen grain and to form the pollen tube. Otherwise, the mature pollen grain probably lost its viability, then it's of no use. So the receptivity of the stigma and the release of the pollen should be what? In sync. Okay, should be in sync. So I'll write synchronization in pollen release and stigma receptivity. Okay, synchronization, pollen release and stigma receptivity. These are the three things that I require. What do I do not require? I generally do not require pollinators. Why? Because they are usually very close to each other. So generally, no pollinators are required. Second thing that you have to notice, and there's a red flag over here regarding this particular thing, which is that no genetic variation is going to be seen. Why? Because the pollen grains are having the genetics of the parent plant, same parent plant as the, the male gamete and the female gamete will be from the same plant, right? That is why there is no genetic variation that is going to happen. So autogamy means self-pollination. For that you need the flower to be bisexual. The flowers have to be close to each other. There should be synchronization in the pollen release and the stigma receptivity. No pollinators are generally required. That is a good part of the story, but there is a bad part, and that is what? No genetic variation. Okay, ma'am. Now the next thing that we have over here is when the pollen grain from this flower comes and lands on the stigma of this particular flower. So here, is it the same flower logic? No. Here this is case 2. Case 2 means basically different flowers, but same plant? Both of them same plant? Yes, same plant. What do you call this as? This is known as, just a second. Yeah, this is known as gytonogamy. Different flowers of the same plant, this is known as gytonogamy. Is it self-pollination? No. Since two flowers are involved over here, not two plants, but still two flowers are involved over here, I'll call this as cross-pollination. Is it true cross-pollination? Ideally not, because it's still in the same flower, in the same plant. So it's a cross-pollination, but not true cross-pollination. We cannot say that. Genetically, if at all, do you think that the progeny is going to be just similar? Yes. Again, the pollen from the same plant, the female gamete from the same plant. No genetic variation. Here also you have the red flag. That is, just give me a second. Yes, that is no genetic variation. This is very, very, very important. Okay, got it? Now, over here, the last scenario. Ma'am, I really want a green flag. I really want a green flag where I see genetic variation. So listen to me carefully. From here, if the pollen grains travel and go there in that particular flower, that's a whole different flower, right? So let's make that. So just make that. This is a third case. And here what is happening? Different flowers. And not just that, different plant. This is a whole green flag. We say no, green forest, because this is actually the true cross-pollination that we have. and it is the only one that brings genetic variation. It's the only one that is going to bring genetic variation. Very, very, very important thing. What do you call this as? This is known as xenogamy. It is going to be known as what? Xenogamy. So the first thing you had autogamy, the second thing that you had is geitonogamy, and the third one is known as xenogamy. In this, only one is self-pollination, both of them are cross-pollination, but this is not true cross-pollination. Why? Because there is no genetic variation happening over here. This is actually cross-pollination, true cross-pollination, where genetic variation is going to be happening. So can I say, functionally this one, okay, geitonogamy is functionally cross-pollination, but genetically is just like autogamy? Yes, genetically it is just going to be like autogamy because there is no genetic variation being seen, but functionally it is cross-pollination because two flowers are being involved over here. There are three things, and for xenogamy you can remember X looks like this, so there is a true cross-pollination happening over here. Do not be confused at all. Okay, this is something that you need to know and is super, super, super important. Now, I'm going to extend this and I'm going to talk about one more very important thing before I go ahead with anything else, and that is going to be. So, in autogamy, while you discuss autogamy, there are two different flowers that is given in your NCERT. Different types of flowers, and it goes like this. So, there is a flower which is open like this. And in the same plant, you might be able to see flowers which are closed like this. Can you guess what's the difference? What is the good part of having the flowers open and having the flowers closed? So listen to me. The open flowers which are there, okay, you have a name for them. You call them as, work, macha, work. Chasmogamous flowers. Okay, what do you call them? Chasmogamous flowers, they are the open ones. Now, what do you call them as? You call them as clistogamous flowers. And what is the use of having clistogamous flowers? I'll talk about it. So, they are closed ones. Now, you only use your logic and tell me, the chasmogamous flowers which are open, do they show autogamy? Yes, ma'am. If this bisexual, yes, it can show autogamy. Do they show geitonogamy? Yes, you have another flower on the same plant, it can show geitonogamy. Do they show xenogamy? Yes, ma'am. Obviously, if you have another plant nearby, it can show xenogamy. So, can I say three types of pollination? Okay, or basically three types of everything is going to happen? Yes, ma'am. What about clistogamous? Clisto, Cl. Remember, they are closed. They are going to be closed. Autogamy, xenogamy, or geitonogamy? Xenogamy, no, it cannot happen, it's closed, ma'am. Geitonogamy also cannot happen. The only thing that can happen is autogamy. Exactly. So, clistogamous flowers, which are the closed ones, Baba, they always show one thing, that is only and only autogamy. Oh my God, Jasmine, have patience. For God's sake, yes, autogamy. Okay, we have only autogamy happening up over here. Cool. And which is why good part is there. Good part is that assured seed setting would be there. Assured seed setting means whatever you do not have to be worried about pollinators or anything. There will be seed being formed. Anyway, pollination is going to happen. Most probably fertilization will also happen. Assured seed setting will be there. You do not need, no requirement, no pollinators required. No pollinators required. Over here, sometimes you could still need, for autogamy also, you could still need pollinators because of the positioning, you might need it. But here in the clistogamous flowers, there is no need, no pollinators required. A short seed setting is there. What's the problem? Genetic variation is going to be nothing. Okay, the same problem about autogamy remains there. There is no genetic variation that is going to be there. Okay, now in your NCERT there is 3 examples of this plant which shows this, and I remember it as this: cow, Commelina oxalis, and there is viola. Commelina oxalis, and there is viola. These three plants are there, and how I remember it as cow with a V, not a W. This is very, very important. Please do remember the names of it. Please do remember that this clistogamous flowers and the chasmogamous flowers could be on the same plant. Usually the clistogamous flowers are inside the soil or probably just about the soil, okay, because they do not need much exposure. Anyway, they are going to be close, so even if nothing there, it's okay, still fine. But sometimes they are within, or sometimes they're just above the soil level. Cool ma'am, done ma'am. Okay, then we come over here and we talk about it. So here we have autogamy. Pollen from anther goes to the stigma of the same flower. Done ma'am. Needs synchrony in pollen release and stigma receptivity? Yes. And also needs bisexual flowers? Yes. Also need the positioning to be done properly? Yes. Okay, the type of flowers, viola, oxalis, commonina, they usually show autogamy. Okay. Chasmogamous flowers are there, open flowers with exposed anthers and stigma, can undergo self or cross pollination. Anything. Autogamy, xenogamy, geitonogamy, anything. Clistogamous flowers, closed flowers. They are always closed. No exposure to external pollen, nothing. Okay, only autogamy occurs. Yes ma'am, there's no option. A short seed set is there even without pollinators. Clistogamy, advantage or disadvantage? If at all you ask me, advantage is seed set is assured. Disadvantage is there is no genetic diversity. Now this is the picture that is given in your NCERT. This is the chasmogamous, and these are the clistogamous flowers. You see they are closed over here. They are closed and they are basically in the root. So actual picture looks like this. Okay, I've taken the actual picture. Sometimes they are above the soil level, sometimes they could be below also. Okay, but they are closed. And they have to be bisexual. Cool. So that's about it. So geitonogamy, pollen to the stigma of another flower on the same plant. Functionally cross-pollination, but genetically like autogamy. Functionally it's cross-pollination, but genetically it is going to be like autogamy because it's the same plant. Xenogamy, pollen to the stigma of different plant. It's a true cross-pollination. Brings genetic diversity. So if you're asked the difference between both of them, write the first point. What do you mean by geitonogamy? First definition of it, that this is in the same plant but different flowers. This is this, this is this. Then you say it is what? Cross-pollination, but not true. Here you say it's a true cross-pollination. Then here you say genetically it's like autogamy, but functionally it's like cross-pollination. Here you say it brings the most genetic diversity. That's all. Three difference, and then that's it. That's all about it. So there's a question. When pollen grains of a flower pollinate the stigma of a flower of another plant, it is going to be xenogamy. It is going to be xenogamy. This is 2025 question. Okay, and now we're going to talk about the agents of pollination. Who does the pollination? Okay, what are the agents? So broadly, if at all I speak, we have two type of pollinators. I'll say there are biotic agents and there is abiotic agents. Okay, you have biotic as well as abiotic agents. The biotic ones that we have is the living ones, is basically the living ones. That is insect. Okay, you have so many insects, so many different types of insects. Apart from it, lizards also sometimes, or lemur, monkeys also sometimes. Unknowingly or knowingly, they do pollination. They go land up somewhere, they move somewhere else. That is how they do it. Abiotic means they do not are living. They are not at all living organisms out there. Right, so we're going to discuss about both of them. What is most common? Obviously biotic. In abiotic, what do you have? You have wind and water. You have wind and water. Since not a lot of them plants are there in the water, you can imagine that wind is the most common in terms of, if you had to compare this and this, it would be wind that is more common. Okay, yes ma'am, done ma'am. Okay, now let's talk about it. The first one is wind pollination, and the question that comes in second PUC is basically, give me the features of a plant that undergoes wind pollination. So you only remember, this is nothing. I always tell this, do not mug this up, understand this. Okay, so over here, when you talk about wind pollination, the pollen grain is there. Just imagine a scenario if the pollen grain is big, large and heavy. Can it walk around? Obviously it becomes very difficult. So which is why the pollen grain has to be light in nature, such that it can easily travel through wind. That is the first feature that is there. Now, the next feature that is there is that it should be non-sticky. Suppose I am the pollen grain, okay, and I am going to the wind. I'll just go stick everywhere out there. Will that be fine? Obviously no. It should be non-sticky. It should only land wherever it is required. So pollen grains which is there, it's light and non-sticky for easy wind transport. Second thing, what about the stamen? The stamen has the anther and the filament, right? And the anther has the pollen grain. If you want the pollen grain to travel through wind, you have to make sure you have it exposed. Okay, it should not be like over here. It should be there where the wind can carry it. So there's a well exposed stamen that is a requirement for the wind pollination to happen. Now what about the stigma? Anyways, pollen grain is not sticky in nature. So there should be someone who is sticky in nature who probably captures it. So suppose I am the stigma. If I am not sticky, the pollen grains will just go off. So I have to make sure that I am sticky in nature. So stigma is going to be not, at least if not sticky, I'll say it should be feathery in nature, such that basically within my feathery stigma, I can just trap the pollen grains out there. So stigma is going to be large, and apart from being large, they have to be feathery to basically trap the pollens out there. Okay, so we studied about the pollen grain, we studied about the stamen, we studied about the stigma, and everything has been done. Now we talk about the flower. Flowers, when you talk about it, many of them are going to come together to form an inflorescence, most probably. But if at all you noticed, inside each flower you will only find that, okay, ovary is there, but inside ovary only one ovule will be there. The reason being that, because it can happen, it may not happen. Why do I make multiple ovules and waste it up? There's no point. Which is why usually you just have single ovule per ovary. There's only single ovule per ovary, just not to waste it. This is a simple logic that you have, please remember it. So many flowers are there, but they will have only single ovule per ovary. This is a unique feature of wind pollination, because we do not know whether it's happening or not. We do not want to waste our energy on things. Okay. Yes. Now I'll just revise it. Wind pollination, the pollen grains, first thing, the pollen grains has to be light and not sticky such that it can travel easily. The stamen has to be well exposed. The stigma has to be feathery and again large to trap this pollen grains. The flowers could be many, but when we talk about flowers, each will be having ovary but only with one ovule. Now example is important in terms of your MCQ. It can be second PUC MCQ, it can be KCET MCQ. The example for wind pollination is grass and the corn cob. Grass and the corn cob. If at all you know, the corn cob over here, okay, if you see this thing that you see, these are the tassels. These tassels are nothing but the stigma and the style. They are nothing but the stigma and the style. So if at all I say that this is the corn that we eat, if inside the corn you have what? You have seen the white part, right? Do you know that basically, this particular thing that comes off, right? This is nothing but the stigma, the style, and the ovary. I told you the ovary which is there, it transforms into fruit, so that is the corn that we eat. Inside that you have ovule, that is the seed, that is a white portion that we eat. If at all fertilization has not taken place, basically you might have seen it forms a white thing only. There is no fruit being formed, it is just flat thing. That is the logic. So this is the stigma and the style that we have, and the example for that is corn cob. Please remember it undergoes wind pollination, and apart from it you also have the grass. Okay, and it has single ovule. Yes, done ma'am. Now we go ahead with the next thing, that is water pollination. In water pollination, please remember, okay, usually it is very rare in flowering plants. The flowering plants which are there, which you call as angiosperms, it is very rare. And if at all you still want to count, yes, it is there in what? In mostly the monocots, monocot, not dicot. In angiosperms you have two types, monocot and dicot, right? Dicot has two cotyledon, monocot has only one. We'll talk about that. You might have studied in 11th grade as well, but just telling you. Okay, so mostly it does not happen in the case of flowering plants, that is angiosperms, but if it happens in monocots, and only 30 genera of monocots. This is important. This is there in your NCERT. Only 30 genera of monocots, basically, they are going to have this type of pollination that is known as water pollination. Cool? Yes. Now example for it. It is actually common. So it is actually common in water pollination. We have studied that the lower plants which are there, they required water for everything. So algae, bryophytes, pteridophytes, they are the lower plants. This is more common in them. They are going to be more common in them. There is no explanation about gymnosperms out there, so you also need not be bothered. Algae, bryophytes and pteridophytes, these are all the lower plants, okay, lower divisions of plants, and all of them mostly show water pollination. But when it comes to angiosperms, they mostly do not show, and if they show, it's usually monocots, and in monocots 30 genera. Okay, now you have the flowers which are there, okay, or basically the plants which shows it. You have to learn their examples. show. There are some plants which are going to be marine, that means they are found in the sea, and there are some plants that are going to be in the fresh water, where the salt content is going to be less. So the example for fresh water is going to be Vallisneria and Hydrilla. Vallisneria and Hydrilla, they are freshwater plants where the salt content is going to be less. And there is an example for marine plant, and that is known as Zostera, or you also call this as the seagrass. You can also call this as the seagrass. This is very, very, very important. Okay? Yes, ma'am. Okay. Now I tell you one very important thing. Over here, let's see the picture of both of them and try to understand it. So Vallisneria is like this. If you see, this is the surface of the water. The female flowers are usually at the top in the water level. What about the male flowers? Male flowers are over here. So they move out from here. They move up over here. Either they release the pollen grains or the flower itself comes up there. What do you understand from this? It's water pollination only, but it usually happens on the surface. In case of Vallisneria, which is this plant, which is given in NCERT, which is a freshwater plant, the pollination happens in water only, but this is going to be surface level pollination that happens. On the other hand, when you talk about the Zostera, which is a seagrass, seagrass means obviously it's in sea, sea is marine, it has more salt content, so it is happening over here. And if you look over here, it's within the water itself. There's no option to reach at the top, they are going to be within the water. So always understand, here in this case, the pollination is going to happen in the submerged condition. In the submerged condition. And the pollen grains which are going to be there, they are going to be what? Ribbon shaped. They are going to be ribbon shaped. This is very, very, very important. So Vallisneria, the female flowers float, the male flowers, or basically the pollen grain, drift to them. They usually show surface level pollination in water. On the other hand, sea grasses such as Zostera, pollination occurs underwater, and here the pollen is going to be long and ribbon like. And do understand, since it is happening in water, usually the pollen grains are covered by a covering that is known as mucilaginous covering, such that no decay is going to happen. Anything that stays in water can get decayed very easily, right? So such that decay is not happening, which is why it is covered by a mucilaginous covering. So pollen protection, mucilaginous covering prevents the wetting out there. So this is about the plants which did it. Vallisneria, Hydrilla, freshwater. Zostera, that is a seagrass, marine. In marine, submerged. In Vallisneria, on the surface. Now, the plants which do not show water pollination, even though they have water in their name, water lily, water hyacinth, water lotus, all of them, those of them who have water in their name, basically they are not going to show water pollination. Instead, they have either wind pollination or they show insect pollination in general, but not water pollination. There are exceptions. Please, please, please remember them. Okay, they do not show water pollination. Cool. So if you're asked about features of a plant that undergoes water pollination, you can just say that they have pollen grains which are usually ribbon-like, which allows them to easily move, or probably they are covered by mucilaginous covering and stuff like that. And then you give examples of both of them and you explain better. Usually they do not ask about the features of water pollination plant because there's no too much thing given about them, right? Okay. Mostly they asked about insect and as well as I said, wind. Okay, now we come to insect, and always understand, or when we talk about biotic agents of pollination, it is never going to be just insects, it could be something else also. So bees, butterflies, flies, beetles, wasps, and all ants and moths are insects, but there are birds. For example, sunbirds, hummingbirds, there are bats, okay, there are primates such as lemur, and there are rodents, there are reptiles such as geckos and lizards, which also does what? Pollination, animal pollination. Okay, and over here, usually in insect pollination, if at all we talk, because they are the general ones, they do this pollination. So over here, the general thing about them is, in insect pollinated flowers, the flowers are going to be very large, they're going to be conspicuous, they are going to be colored, they're going to be fragrant in nature, such that they can attract the insects out there. Insects cannot see the colors that we see, but they see things differently, which is why it has to be still large and colorful. Okay, now, if at all you talk about flowers, sometimes they can be grouped up together to form an inflorescence. So small flowers, if at all it's there, they come together to form inflorescence. Usually the flowers are large, but if at all you're talking about small flowers, they do not remain small, they come together to form an inflorescence so that they look big. And basically pollination can happen easily. Now in insects also you have flies that does not like good smell, but they like bad smell. So they have a very foul odor. The plants that undergoes insect pollination via a fly or a beetle usually has a foul odor. You might have heard about Rafflesia. So very big flower, it smells so bad. Okay, so yes, remember that. And the next thing that is super important is the sticky pollen grains. Remember, the flowers anyway cannot move. Okay, they cannot do anything. So any insect that comes, for example, I am an insect, I came to drink the nectar. What is the plant going to do? Basically, since the pollen grains which I'm going to produce is going to be sticky, it's super easy for the pollen grains to stuck on my surface. And now I'll be going to some other plant and basically unknowingly I'm going to check these pollen grains. So this is very important that the pollen grains over here has to be sticky. There in wind it was not sticky. Here in insect pollination, it has to be sticky. Okay, so insect pollinated flower, either large and colorful and blah blah blah. Small flowers if are there, they are coming together to form inflorescence, to form the visibility. Fly and beetle pollinated flowers, they secrete foul odor such that they can get attracted easily, and sticky pollen grains are there to facilitate the pollen transfer. Apart from it, one more thing, that is they give floral rewards. The plants wants the insect to come back, visit again and again, which is why floral rewards are given. This is also a question, second PUC question. So floral rewards as what? Nectar. First thing, they provide nectar. Second thing, they provide pollen grains. So you should not think, ma'am, but pollen grain has to go and help in the process of the pollination, fertilization later on, right? Only few pollen grains they will eat, rest of them will be there on the surface. So it's not like they are selfish creatures eating everything, it will be there. So nectar and pollen, these are something that they give. Apart from it, they also sometimes provide safe places for laying their eggs, for the egg of the insect. Yes, you know what? You have seen this plant? This flower that you see, these are 3 human beings, which I think has more height than me, and still this flower is very tall. This is Amorphophallus, and obviously the organisms that come over here that does pollination and all, they can provide a little space for this particular flower, can provide a little space for these organisms to lay their eggs. So they do that. They are going to do that. Okay, please do know that. And some of these Amorphophallus flowers are smelling so bad, some of them, not all of them, such that flies and beetles could be attracted. So what are the rewards? If at all you ask me, nectar, and apart from that pollen, and apart from that safe places for laying the egg. Now, there's a special relation that is given in your NCERT which blew my mind when I read about it, and that was this. So this is yucca plant and this is moth. Both of them look the same, right? Yes. So what does the moth do? You know what? Both of them cannot live without each other. Okay. So these guys which are there, they mate. Okay, the moth, they mate and they come and they go and lay their eggs where? Inside the ovary of this particular plant. Inside the ovary, they make a small chamber and get inside and probably lay their eggs there. Okay. And then they come back in search of food, because obviously they have to feed their young ones. They come to the flower and again take the pollen grains. They take some pollen grains, they keep it like this. But they keep some of the pollen grains there also. Anyways, they take some pollen grains, go back inside the ovary and give it to the small little ones which are inside. And you know what? One thing that they do, they come back and they put the pollen grains from here and transfer it to some other plant also, such that easy pollination can happen and this can sustain. They have their home. If at all a plant is not undergoing pollination and this fertilization is not there, basically the flower is not going to come up. So this is what it usually happens, and I was actually very, very surprised to see all of it. So remember about yucca and the moth, they show obligate relationship, means that they cannot live without each other. Okay, and here the moth is provided a safe place to lay the egg, and the moth is actually helping the plant for pollination as well. Okay, so Amorphophallus, and this is the example that you need to learn. And yeah, what type of questions usually come from this particular topic? This type of question. Picture related to pollination is given below. Answer this question. So what is the picture? Insect is there and a plant is there. Will this pollination confirm fertilization? Justify your answer with the suitable reasons. Obviously no. As I told you, all pollination does not confirm fertilization, because after pollination, for the process of fertilization to happen, you explained that pollen tube has to be formed. Later on, the pollen tube, after being formed, it has to reach the ovule. Inside that, you had to go to the embryo sac and fuse, basically, and then only we can say fertilization has taken place. So obviously pollination does not confirm fertilization. Pollination only brings pollen grains to the stigma. It is just a transfer process. Fertilization occurs only if pollen is compatible, germinates on the stigma and forms a pollen tube. So every pollen that lands over here does not form the pollen tube. Know that. Please know that. Okay. Next is, what are the floral rewards? I told you nectar, pollen and the safe place to lay the eggs. Mention the characteristics of flower in this pollination. I told you about all the features. Please remember that. That's all. There's a 5-marker question, you can get straight 5 marks. Now, which of the following aquatic plant does not show pollination by water? 2021 KCET question. Does not show water pollination. Vallisneria, hydrilla, water hyacinth, zostera. You know, water term comes, it does not show water pollination. Water hyacinth is the odd one out. It does not show water pollination. It usually shows wind or insect pollination. Cool, ma'am. Cool, ma'am. Now, we're going ahead with a new thing out there. I hope pollination part is also clear. We're going ahead with the next thing, that is outbreeding devices and artificial hybridization. This is going to be super important, so make sure you listen to me very, very, very carefully. What do you mean by outbreeding devices? Okay. Outbreeding means, you know what, I told you, if self-pollination is happening again and again, suppose I have a toxic character and my pollen grains are landing on the stigma of my plant only, what is going to happen? The same type of me, toxic characters are going to go ahead, nothing good. Will the plant be able to survive later on, the progenies? No. This is known as inbreeding depression. Because of continuous breeding within themselves, what is going to happen? There's a depression in a plant that is going to take place. Even plants are depressed. Now talk about human beings. Right? To avoid this, plant itself has thought that I am going to have some outbreeding devices which can promote cross-pollination. Not self-pollination, I want to promote cross-pollination. So plant came up with an idea. Plant is a very shy person. Okay, so over here, the first idea is, let's have some time difference. Let the pollen release, but the stigma is not going to be receptive. The pollen is released, but the stigma is not receptive. It's of no use. It's of no use. So in a bisexual flower also, the pollen is there, the stigma is there. The pollen grain itself has come, but the stigma is not receptive to receive it. Is there of any use? No. Obviously, this will promote cross-pollination. That is the first point. Second point, spatial separation. What do you mean by that? Spatial separation basically means, suppose the, just a second. The female part, it looks like this. The male part is over here small. From here to here, it has to go, it becomes very, very difficult. It can be even smaller than that. This is known as spatial separation. Anther and the stigma are positioned very differently, such that pollination does not happen. Second thing, do not mug it up. Imagine. Time difference, the second one is spatial separation. The third one is self-incompatibility. Now, this is something that is genetic mechanism. Is the same plant, is the same thing, everything. The pollen comes and lands. It could have formed the pollen tube, but somehow, because of some genetic mechanism, it does not allow pollen tube growth to happen, does not allow pollen germination to happen. This is known as self-incompatibility, and this is what a genetic mechanism that prevents self-pollen from fertilizing the ovules. It does not allow the pollen germination itself to take place over here. This is what self-incompatibility. Time difference, spatial separation, self-incompatibility, incompatibility. The last one is going to be unisexual flowers, production of unisexual flowers. So here you have two types of flowers, monoecious and dioecious. Okay, you have two types of flowers, monoecious and it is going to be dioecious. What's the use of it, ma'am? I do not understand. For this, I'll require some space. Listen to me very carefully. So when I say unisexual flowers, what do I mean by that? A flower just has one sex. Now, suppose this is a plant. Here, all flowers are female. Okay, autogamy can happen. Geitonogamy can happen. No. Why? Because you do not have the male part anywhere. Here, you only have flowers that has the male part. This is another plant. What does it promote? It promotes one thing, and that is xenogamy. This type of plant are known as dioecious plant. They are known as dioecious plant, which has basically male plant and female plant. The flowers are unisexual and the plant in total is also unisexual. We say male and the female plant. This is dioecious plant, and the example for this is papaya. Is what? Papaya. So in papaya, there will be a plant that will be having, so I've produced a picture over here. Female flowers will be there only on this particular plant, male flowers only on this particular plant. What does it promote? Xenogamy, cross-pollination, true cross-pollination. These are dioecious plants. Now, they have also mentioned one more thing, and the example is important, okay, papaya. They've also mentioned about monoecious plant. What do you mean by monoecious plant? Monoecious means, again, you have to make sure they are unisexual, right? So over here, the flowers can be like this. One is a female and one is going to be male. So for monoecious, for them to be monoecious, the rule is that both the sexes should be on the same plant. It can be either like this, or sometimes it can also be like this, where you have both the sexes on the same plant. But we're not talking about monoecious which is like this. We're talking about monoecious plants having unisexual flowers. We have to be very specific there. So some monoecious plants have unisexual flowers, and in them autogamy will definitely be prevented, but gytenogamy will still be there. Well, gytenogamy will still be there. But anyway, in this particular case, it mostly helps in gytenogamy or it helps in xenogamy. Okay, autogamy is prevented for sure. At least one thing is prevented. No, that is why we have written about that. So always remember, this is dioecious, means two separate sexes in two separate plants. The flowers definitely will be unisexual. Here in monoecious, the flowers can be bisexual or unisexual, but I'm talking about unisexuality. So I'm talking about this condition. The flowers, suppose they are like this, what is going to happen? They prevent autogamy. This is important, but not gytenogamy. Gytenogamy can still happen. Okay, prevents autogamy only. Gytenogamy can still happen. Xenogamy will definitely happen. Okay. Example, castor and maize. Example, castor and maize. I remember it as Chief Minister, CM. Castor and maize. Okay. So here, monoecious unisexual flowers, monoecious example castor and maize, male and female flowers on the same plant, prevents autogamy but not gytenogamy. Dioecious example papaya, male and female flowers on different plants, prevents both autogamy as well as gytenogamy, only promotes xenogamy. So what are the four things? Time difference, spatial separation, self-incompatibility, and the final one is production of unisexual flowers. By the way, castor oil is something that we put on our hair. Okay, this is the castor plant. It has both unisexual flowers, male and the female on the same plant. This is outbreeding devices. Again, you can get question in second PUC based on this. Questions have come on the basis of this. Important. This is important. Okay. Now, guys, do you want the PDF of this? If you want, please let me know in the comment section. Just a second. Yes, perfect. Now I go ahead with the question. So in some plants, the stigma and the anther mature at different times. Because of what? They are maturing at different time difference. It attracts pollinators? No. It facilitates self-pollination? This definitely, it's not helping in self-pollination. No. It prevents cross-pollination? No. It is facilitating cross-pollination. This is your answer. This is 2019 question. Okay? Okay. Now, we come up with the next thing, that is artificial hybridization. What do you mean by artificial hybridization? It's a very, very important thing. It means, first of all, it's a part of crop improvement program. So I remember previous year question came, and it's a 5-marker question which came. It said that, please mention about the crop improvement program which helped the farmers to produce superior variety of crops. I always say this, when you go ahead with all the chapters in second PUC, especially the biotech ones, you will have a confusion with this question. The reason being that there also you produce genetically verified crops, modified crops. Here also you're doing it, but here it's not genetically modified. That's the logic. This is a part of crop improvement program where actually farmers can produce superior variety of crops without do any genetic mismatch out there, or probably genetic changes. Here what you're doing is, suppose I am a flower, and over here there is male and the female part. If I let the female and the male part be there, what is going to happen? From the male part, the pollen grains are landing on the female part and the pollination will happen naturally. We do not want that to happen. Why? Because I have looked for, I'm a farmer now, I'm looking for another plant. This plant that I've looked is having good feature, is just pest resistant. It is resistant to pest. That plant that I'm looking for is basically drought resistant. So two good features are there, and I just have to fuse them up. How can I do that? So the first process is that if that flower is bisexual, please remember, only if that flower is going to be bisexual, that means it has both the male and the female parts, I'm going to remove the male part from here. Why? Because I do not want the pollen grains to land. This is known as emasculation. What do I call it? Emasculation. Very, very, very important. Emasculation. Okay, the masculine part, I'm removing it. Cool. Now after doing that, what do? I just cover it up. This is known as bagging. After removing it, I cover it up. Why do I cover it up? Question should come in your head. Why? Because I do not want any other pollen grains to come and land over this, because I have made sure that that plant that I am looking for, I'll take that pollen grain myself. This needs a human interference and I am going to put it there. So I just bag it up. Why? Because I am waiting for the stigma to become receptive, let it become mature, and I'm just keeping it there. Then one day, I collect the pollen grains. So this was the first step. The second step. The third step will be collection of pollen grains from the desired plants. Pollen grain from the desired plant. Okay, now you have got the pollen grains from the desired plant. What do? You open this and you put it up. Okay, transfer of pollen grains. Next one will be transfer of pollen. After doing that, what I'm going to do? Again, I'll bag it up. Why? Because again contamination can happen, any other pollen grains can come. So this is now known as rebagging. I'm doing it again, that is what is known as rebagging. And the last one will be tagging, where I'm tagging that which plant is the one that I have fused together, so that I have an understanding of it. Please understand, this requires human intervention. This is part of crop improvement program to produce superior variety of crops out there. This is known as artificial hybridization because we are artificially doing it, man-made. And it is also known as controlled pollination. Controlled pollination. Why controlled pollination? Because we are the one who is controlling the pollination over here. We make sure that we carry the pollen grains and we put it over here. It's happening in a very controlled position. It's not happening naturally. That's why. Controlled pollination, artificial hybridization, all the same thing. First is emasculation, only if it's a bisexual flower. If it's a unisexual flower, you do not need emasculation. You do not need to remove the male part because it's not there. Second one will be bagging. You cover it up. Then the third one will be collection of the pollen grains from the desired plant. Then you will transfer it up. Okay, artificial pollination is going to happen. Then the fifth one is going to be rebagging. Again, I'll cover and then I'll tag it up. That is known as tagging. These are the steps that you need to remember. Okay? Yes, ma'am. Done. Now, plan an experiment and prepare a flow chart of the steps that you would follow to ensure that the seeds are formed only from the desired sets of pollen grains. So this is the question. Plan an experiment and prepare a flow chart of the steps that you would follow to ensure that the seeds are formed only from the desired set of pollen grains. This is what? Controlled pollination. Now don't think that we studied right now, that's why you know. But when it comes in the question paper, you do not know where it is coming from. So desired sets of pollen grains, just understand controlled pollination. Name the type of experiment, artificial hybridization, controlled pollination. Write the importance of the experiment. What is the importance? Basically, I get to choose the plants for desired characters and I can produce hybrid varieties of crops out there. And that can sustain in any environment, any nature out there easily. It's a 5-marker question. Know that. It's very important. So, yes, go step by step, explain it properly and you get the 5 marks. Importance of such experiments: they prevent contamination by unwanted pollen and help in producing hybrids with desired characters such as higher yield, disease resistance, better quality, etc., which is essential for crop improvement program. Now, identify the correct order of steps involved in artificial hybridization. KCET question. That is 2019 question. Correct step. First one is going to be emasculation, so I just have to look for that, emasculation. Then bagging, correct? Yes. Then artificial pollination, yes. And then rebagging. Right? So this is going to be the answer. If you want to look forward and just check out all the options, you can. My energy is drooping down because it's been a long time now. Just give me a second. I hope you have energy, high on energy, right? Those of you who probably are gonna watch this video later on, and I'm not sure because I feel that in this channel, in our channel, I have seen the trend. Only during examination, everyone comes and says, oh my god, ma'am, I should have discovered this channel first. But somehow, you get lost now. So if you're watching this video and if you feel that, okay, this session is actually nice, share this channel with all of your friends, hit on the subscribe button and tell them to attend the classes. Okay? Yes. Cool. Now only if you feel so. Now going ahead with pollen pistil interaction. We studied about pollination. Suppose the pollen grain has landed on the pistil. What's the next step that is going to happen? I'm going to talk about that right now. And then we'll talk about fertilization. So over here, we are almost up there. We're almost going to finish, okay? 30 more minutes, I think so. Anyways. So here you have got your pollen grain. First thing that you need to know is that here there is going to be some chemical interactions. Okay, and if all goes well, I'll say that it is a compatible interaction. If all does not go well, I'll say it is incompatible. If it's compatible, what is going to happen? If incompatible, obviously it is not going to germinate. Okay, if it is going to be, just a second, if it is going to be incompatible, the next thing that you're going to see is that there won't be no germination. Got it? So over here, if it's compatible, the pollen grain is going to land over here, make the pollen tube like this. Will it make the pollen tube? Let's check it out. Just give me a second. Yes, it makes the pollen tube. And then what? And then over here, you have the ovule. Inside the ovule, what do you have? You have the embryo sac. Inside the ovule, you have the embryo sac. Here you have the synergids. Here you have the female gamete. Here you have the antipodal cells, everything. So basically, when you have got the pollen tube over here, the pollen tube is going to come over here. First thing that it's going to do is enter the ovule. First, it enters the ovule. Second, it is going to enter the embryo sac. And later, the male gametes which are going to come along, they are going to fuse. I'll tell why you have two male gametes and stuff like that. If you remember, pollen grains have usually two male gametes, but there's only one female gamete. So what happens? We'll talk about it in the next part. But this is the process that happens. Listen to me very carefully. I'm repeating it again. The chemical interactions happens. If it is compatible, in that case, pollen tube is going to be formed. With the male gametes, it is going to come. First, it gets entry into the ovule, then the embryo sac. Then the fusion and everything happens. If it is incompatible, no germination is going to be seen. And always remember, it's not like only one pollen grain comes and forms the pollen tube. There can be multiple of them that can form the pollen tube, but usually only one gets in. So pollen tube, even after pollen tube formation, there are chances that fertilization will not happen. This does not guarantee it. Okay, this is not guaranteed. That's why I said pollination does not guarantee fertilization. It can form the pollen tube, it can stay just until here and does not go forward. So know that. Now, one more very important thing. If you remember, there was 60% of plant, which basically angiosperms, which release their pollen grains in two cell stage. One vegetative and one generative. And there were 40% of them which send their pollen grains in three cell stage. One vegetative and two generative. Right? In this case, it's all cool because you have, not two generative, two gametes, male gametes. Okay. In this case, it's totally fine. Why? Because the vegetative cell helps in the pollen tube formation and these two gametes can easily travel through. But in this case, if it comes, what is going to happen? Once they reach and everything is okay and it forms the pollen tube, that's when the division finally takes place and you get the two male gametes right over here. Okay, remember that. Once they come land, it's compatible, they start forming the pollen tube, that's when the division happens, you get the two male gametes. But anyway, you will get it. Two male gametes is going to be there for sure. Okay, so pollen pistil interaction, it's a dynamic process involving recognition of pollen by the pistil and either promotion or inhibition of its growth. Either it allows, either it does not allow. Pollination and germination. After the compatible pollination, the pollen grain germinates on the stigma. First of all, please know there's a chemical interaction that is happening. Remember that. It produces a pollen tube through one of its germ pore. Through one of its germ pore, I told you it is going to make a pollen tube. In plants where pollen is shed at two cell stage, the generative cell divides during the tube growth to form the male gametes. So when the tube is growing, that's when, if at all you see over here, okay, when the pollen tube is going to grow, this part, the vegetative cell contributes to cytoplasm of the pollen tube. That's when the generative cell is going to divide into two gametes. Okay, and what is a tube nucleus? Tube nucleus is nothing but basically nucleus of the vegetative cell. And over here you see generative cell nucleus is gonna divide. It's gonna divide. Let's see. It's gonna divide and you have got two male gametes and the tube nucleus is also there. That's a separate thing. Okay. Now, yes. So in plants with three cell stage, the pollen already contains two male gametes, so that's not a problem at all. Now entry into the ovule. The pollen tube enters the ovule through the micropyle. That is the opening in the ovule. It then enters one of the synergids via the filiform apparatus, which guides the pollen tube into the ovule. I always tell you over here, the synergids which are there, they have the filiform apparatus. And why do you have the filiform apparatus? For guiding the pollen tube. Do not forget it. And during this process, one of the synergids, they get degenerated also. I hope this is clear. This is pollen-pistil interaction, and you have got questions like this on the basis of it in KCET. Identify the correct order of events in pollen-pistil interaction from the options given below. Release of male gametes into the embryo sac, deposition of pollen grains on the stigma, entry of pollen tube into the embryo sac, development of pollen tube, entry of pollen tube into the ovule. Now you have been asked the correct order of pollen-pistil interaction. The correct one, the first one is going to be deposition of pollen grains. Yes. And what is that? That is the second point. Deposition of pollen grains on the stigma. Right? So here you have to look for the option that starts with second one. This is the one, this is the one. Remove the other ones. Now, read your options through this. You do not have to go this and then this. Okay, read this and then tally with this. After second, there is fourth. After deposition of pollen grains, development of pollen tube. Is that true? Yes. There is development of pollen tube. After that, what happens? Entry of pollen tube into the ovule. After development of pollen tube, does it enter the ovule? Yes, it enters the ovule. After it enters the ovule, does it enter the embryo sac? The next one, that is third one. Entry of pollen tube into the embryo sac, that is also correct. And lastly, release of male gametes into the embryo sac, that is also correct. Okay, so this is very, very important. Please make sure you study this. Cool. If you see over here, if I go by that only, first deposition of pollen grains, then you have fourth, development of pollen tube, then you have third, that is entry of pollen tube into the embryo sac. Arey Baba, you have to enter it into the ovule first, no? So that is a differentiator point that makes it wrong. Works? Yes. And now we talk about double fertilization. It's going to be super important, so listen to me carefully. Very, 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 very carefully. In double fertilization, what do you have? Over here, you have the antipodal cells with their nucleus, haploid, haploid, haploid. You have the synergids over here with their nucleus, haploid, haploid. You have the egg cell over here with its nucleus, haploid. And I'm going to write it egg cell. And what is it? It's going to be haploid in nature. Finally, what do you have over here? This is the central cell that you have. And this has two polar nuclei, which is why it is diploid in nature. Not haploid, but diploid. Now what happens? So the pollen tube comes. It enters through the micropyle, right? One of the synergids goes out to get degenerated. It helps, filiform apparatus is helping there. One male gamete, you have two male gametes. Out of that, one male gamete. This male gamete is also haploid, if you remember, microsporogenesis. This male gamete which is there, it is also going to be haploid. So this is the male gamete that we have. Come on. Okay. This is the male gamete which is there. It is going to be haploid. Okay. They fuse together to form what? To form a zygote. Agree? Male gamete, female gamete fuses together to form a zygote. That's the bottom line. That's why we did sexual reproduction over here, right? Haploid, haploid. N and N becomes what? It becomes diploid. We started off with diploid plant cells which underwent meiosis. And now finally we have got the zygote which is going to be what? Diploid in nature. And tada, half of work is done. But we have one more male gamete. What happens with that? Let's check it out. The next male gamete comes over here, fuses with the central cell. And this gamete is also haploid in nature. This fuses with the central cell which is diploid. Why is it diploid? Because you have two nucleus already. That is why. N plus N. Now you have male gamete which is also carrying nucleus having N haploid number of chromosomes. So this and this comes together, 2n plus 3n becomes what? 3n. This is what? Triploid. And what is this thing that is being formed over here? This cell which is being formed over here is super important for every one of us to remember. First you got the zygote. The second thing that you get over here, which is triploid in nature, is the primary endosperm cell. It's the primary endosperm cell. It's a cell that is going to be formed with a fusion of this and this, and that's when you get a triploid cell. But please understand, what is triploid over here? The nucleus. This male gamete's nucleus and these two nucleus comes together. That's when you get a triploid nucleus right now. You're going to get a triploid nucleus. And what is this triploid nucleus going to be known as? Nucleus, not the cell. The cell is known as primary endosperm cell. The name of the nucleus is what? The primary endosperm nucleus. It is known as the primary endosperm nucleus, that is PEN. Very important. Okay, now one question. Ma'am, what do you call this and what do you call this? Anyway, there are two fertilization that is happening. Yes, that's why you say double fertilization. But you have separate terms for it. Okay, and I'm going to write in blue. This thing that is happening for the first year, okay, this is known as what? This is known as syngamy. This is where you get the zygote. This thing that is happening over here is known as triple fusion. Why? Because three nucleus are fusing together. So you have syngamy and triple fusion, both of them happening inside the embryo sac, which is why you call this as double fertilization. Very important thing. After fertilization, which cells change their ploidy? Egg cell changed their ploidy. It was haploid, now it becomes diploid. Why? Because it has formed the zygote with the help of the male gamete. Central cell changed its ploidy. The central cell which was there, now it's not central cell anymore, it's known as primary endosperm cell, and it is going to be triploid. It was diploid first, it becomes triploid. What about the antipodals and the synergids? They remain the same. Only changes happening between these two things. Always remember that. This is double fertilization and this is the end of the story. So over here about double fertilization. Release of male gametes. After entering one of the synergids, the pollen tube releases two male gametes into the cytoplasm. Syngamy, first fertilization. One male gamete fuses with the egg cell. This fusion is known as syngamy. It is helping in the formation of a diploid zygote. Done. Triple fusion, second fertilization. The other male gamete fuses with the two polar nuclei in the central cell. This fusion has three haploid nuclei forms. Okay, this fusion of three haploid nuclei forms a triploid primary endosperm nucleus. This process is known as what? Triple fusion. Done, ma'am. Cool then. Everything is cool. Since both syngamy and triple fusion occurs in the same embryo sac, the process is now known as double fertilization. This is a unique feature of what? Flowering plants. This is not seen in gymnosperms or things like that. Only in angiosperms you could see this. Now post-fertilization development. The central cell after triple fusion becomes the primary endosperm cell. I told you, and basically and develops into the endosperm. I'm coming to that. And the zygote develops into embryo. Listen to me very carefully. So now you have got the primary endosperm cell and you have got the zygote. Okay, you have got two things over here, primary endosperm cell, and then you have got the zygote. Zygote is what? A single cell made by the fusion of two. This zygote have to transform into an embryo, which is going to be multicellular. For that, it needs nourishment. Who provides it? So the primary endosperm cell which is there, it is going to undergo division and it is going to form a tissue which is known as endosperm. First this happens, this transition is going to happen, then only zygote to embryo transformation is going to happen. Why? Because you need someone to nourish it, and that is going to be endosperm. So always remember, endosperm development precedes the embryo development, or basically endosperm development is the first thing that happens, after which you have the embryo development. Very important. Also, one more change that is happening over here is that you had the ovary, you had the ovule. Now the ovule has basically had the embryo sac which underwent fertilization, which is why this fertilized ovule, which has the zygote and all, is now known as what? It is now known as seed. And what about the ovary? Ovary will now turn into a fruit. This is after fertilization. Okay, this is after fertilization. Cool. Done? Yes, ma'am. Now we're going to see first how this transformation happens, then we will see this. Okay. Okay, so in apple, the chromosome number of gamete is 17. What is the chromosome number in its primary endosperm nucleus? Can you give me the answer? The number of chromosome in the gamete is 17. Always remember, gamete is going to have only half of it, N, in a gamete. Talking about primary endosperm nucleus. Primary endosperm nucleus is what? Triploid in nature. You studied that. By 3 nucleus, okay, 2 polar nuclei and 1 male gamete. Okay, so if N is 17, then 3N is equals to what? 3N is equals to 3 into 17. That means it becomes 51. The answer is going to be 51. You get this type of KCET question from this topic. Okay? Okay, ma'am. Perfect. Now, double fertilization. Okay. I think I'll just skip that question for right now. Yes. Otherwise, it is going to be too late. Okay, so probably, okay, I'll come to that. There's a question that I actually want you guys to solve and let me know the answer. This is a question that is there. Double fertilization is the unique feature of angiosperms, and the products of this double fertilization are zygote and PEN. Zygote and primary endosperm nucleus. In the context of this, when a hexaploid plant, okay, when a hexaploid plant is pollinated by a tetraploid plant, find out the ploidy of zygote and PEN through a schematic representation. Can you try? Hexaploid plant and a tetraploid plant. That is the question over here. Listen to me carefully. I'll tell you. Okay. When you're talking about a hexaploid plant, what do you mean by that? You mean that it is 6N in nature. When you say tetraploid, you mean it's going to be 4N in nature, right? Okay, now who is what? How do we know who is the male, who is the female? So, hexaploid plant is pollinated by a tetraploid plant. That means this is going to be a male plant. And this, on the other hand, is going to be a female plant. Okay, now we have to fuse them and show what is going to happen. Listen to me. If this is a normal ploidy of a cell, when you talk about gametes, it is going to become half. The gamete is going to become half. So if it was 6N before, what happens in the gamete? If a normal cell is 6N, what happens to the gamete? It becomes half of 6N. And what is it? It becomes 3N. Gamete, the female gamete, is going to be what? 3N in nature. What about the male gamete? Male normal cell, again, is 4N. What about the gamete? It becomes half of 4N, and that is going to be what? 2N. Now, how do you form the zygote out there? For the zygote, this gamete and this gamete has to come together, male and the female gamete. Male and the female gamete has to come together. So 3N plus 2N becomes what? 5N. It becomes pentaploid. It becomes pentaploid. What are you talking about? You are talking about the zygote. The zygote. Okay. Now, what about the primary endosperm nucleus? Do you have anything to talk about the primary endosperm nucleus? So over here, in this particular case, the primary endosperm nucleus which is there, okay, it is formed by the fusion of what? 2 polar nuclei. Polar nuclei will also have whatever half the number of, so polar nuclei is there in the case of females. So one nuclei will be having the same number of chromosomes just like a gamete, right? So it is 3N over here. Another nuclei will also be 3N. Together they become 6N. That is a different thing. But primary endosperm nucleus is made up of 2 polar nuclei plus what? Plus the male gamete. What is a male gamete? It is 2N. It is what? It is going to be 2N. It is going to be 2N. So 3 plus 3, 6, 6 plus 2, 8. 8N, that is what? Octaploid. It is going to be octaploid. What is the ploidy of this of? This is our primary endosperm nucleus. So yes, you have to be careful when you talk about male and female, and you should know primary endosperm nucleus contains 2 polar nuclei of a female. So you have to consider this. So this is a general thing. And yes, if you want a whole answer, it goes like this. You can probably read it, take a screenshot of it right now, and yes, it could help you. Cool, ma'am. Now we go all about the endosperm as well as the embryo. Listen to me very carefully. We have got the primary endosperm cell. From that, we have to form the whole endosperm as a tissue. How do we do that? So, this is the primary endosperm cell, suppose. And basically there's a nucleus, primary endosperm nucleus. How it is? It is going to be triploid in nature. This nucleus, first of all, it undergoes division. It undergoes division, divides so much. And these divisions are going to be what? Free nuclear division. What division is it? Free nuclear division. And mitosis or meiosis? Mitosis. The number of chromosomes remain the same. So it was 3n, it remains 3n only. So when the free nuclear division happens, so there will be so many nuclei like this. 3n, 3n, 3n. Okay. What happens to the next ones? There are some other nuclei which is there, which is also 3n, 3n, 3n. But these are covered by cell wall. They are going to be covered by cell wall like this. Please know this. Very, very important. So, ma'am, what are you trying to say? I don't get it. So, I am telling you, nuclear division happens. This is free nuclear division. Cell wall is not constantly forming. When cell wall forms, some of them immediately get their cell wall. Some of them do not get their cell wall. The ones that get their cell wall, they are going to come up together to form the endosperm tissue. You might have seen that in coconut. In coconut, the white portion that we actually eat is where these cells have already been formed. So the cells are grouped up together like this. They have already formed there together. Okay, they are all going to be together. They have their own separate nucleus, blah, blah, blah. They have cellularized. But there will be still some nucleus which have not underwent cellularization. That means cell wall formation did not take place out there. They are going to be free nuclear in condition still. And that is found in the water of the coconut. So the water that we drink is nothing but the free nucleuses which are floating out there. Slowly, as in when the plant matures, what is going to happen? These nucleus is also going to be covered by the cell wall and it becomes more thicker. The kernel is going to become more thicker. It takes time, but it happens. It takes time, but it happens, which is why when we go have coconut water, in some of them we see that there is no white portion because all of it is in free nuclear condition. But in some of them, if it's a older one, what is going to happen? You will see the white one is thickly being formed. Why? Because cellularization has taken place already. Cool. And when this endosperm is completely being formed, after that there's a zygote also. That zygote is going to transform into an embryo. That zygote divides further to form the embryo. But before that, the endosperm has to form. So endosperm is basically a tissue that is being formed with the help of the primary endosperm cells out there. Cool. Yes. So let's see. Why does endosperm develop first? Because it wants to provide nutrition to the developing embryo. Okay. Process is what? Primary endosperm nucleus divides repeatedly, forms triploid endosperm tissue. It divides repeatedly, forms what? Triploid endosperm tissue. All the cells, all the nucleus are triploid. That is why triploid endosperm tissue. Now this free nuclear endosperm, primary endosperm nucleus undergoes nuclear division without cell wall formation. Without cell wall formation, they are just going to divide. Then later on, cellularization happens. Cell wall forms, making the endosperm cellular. Later on, what is going to happen? Cell wall formation is going to happen and it is going to become cellular. Okay, that's how you get the white kernel out there. Then you talk about fate of endosperm. I'll be talking about it a little late. So fate of endosperm is that sometimes the endosperm which is being formed over here is completely utilized by the embryo. Sometimes in some of the plants, the endosperm is left behind as well. So that is two different scenarios and we will be talking about that later on. I'll be talking about it, but it's not the correct time. Okay. So please do understand that how does the endosperm form now? We have got the endosperm. Now we are going to go ahead and talk about what? Talk about this thing that is the embryo. That is from the zygote, we have to form the embryo. All of it is happening inside the ovule. So listen to me very carefully. In the case of plants, we have two types of plants in angiosperms, the flowering plants, monocots and the dicots. Monocots and the dicots, all of them has the same initial step. There's a zygote which is single celled, it divides into two, it is going to divide into two. Okay, this is known as pro-embryo. Further, the pro-embryo becomes globular embryo, the globular embryo becomes heart-shaped embryo, and then it becomes a mature embryo. And you are going to be asked about the sequence of it. So how do I remember it? Okay, pro-embryo, I have to remember it as such. Zygote becomes pro-embryo. Then after the pro-embryo, you know, when we are chotu people, we eat so much, okay, and then we become golu-molu. So globular. The globular. Then we evolve better, we have some crushes out there. So it is heart-shaped embryo. Then when we mature, we know that it's all of no use, it's a breakup thing. So that's a mature thing. Or basically after the heart-shaped forms, we get a breakup and then we become mature. So zygote, then pro-embryo is there, then globular embryo is there, okay, then heart-shaped is there, and then finally you are going to have the mature embryo. This is common as the first initial steps that you have. Okay, the first initial steps that you have from the zygote to embryo formation. Remember, before this, the endosperm has already been formed. The steps is important. Do not miss out any one of them. Super important. Okay, now talking about the embryo, I told you there are two types of plants, dicot and monocot. Dicot will be having a different embryo, monocot is going to have a different embryo. And let me just drink water because it's 2 hours and I'm thirsty. Ready, Macha. Okay, let's go. So in the dicot embryo, what all do you have? And what all do you need to know? Listen to me very carefully. I'm going to tell about the story, okay? Only if I tell the story, then only you will remember it. That's why. I told you, in some embryo, they take up all the endosperm. That's the case over here. Okay, so it's just like having a baby. So over here, this baby which is being formed, it says that, you know, I have grown up, I need to have my own things, I cannot be dependent on my parents. I'm not like that. So it's just like that. The dicot is going to be like that. It takes up everything from the endosperm and it makes its own reserve food material. Yes, and that is known as cotyledons. In this case, it has 2 cotyledons. It has how many? 2 cotyledons, which is why I'll call this as a dicot. And for that, did it utilize all the endosperm which was left behind? Yes. So endosperm completely utilized to form what? To form the cotyledons. So they have dicot as in 2 cotyledons. Now over here, listen to me very carefully. This portion which is there below the attachment of cotyledons out there. Okay, this portion which is below the attachment of cotyledon, what do you call this as? It's known as hypocotyl region. This is known as hypocotyl. This region which is above the cotyledons, above the attachment of cotyledons, what do you call this as? This is known as epicotyl region. Now, first of all, these are 2 cotyledons, but what is this, ma'am? This is the embryonal axis. Actually, the true embryo which is going to form later on into a whole plant. Okay, so this is what? This particular portion that you see overall from here to here is actually known as the embryonal axis, which has epicotyl and hypocotyl. At the epicotyl, here you have the portion that forms the shoot later on, as in the leaves, the stem and everything later on. It is known as the plumule. What do you call this as? It is known as, just a second, the plumule. Okay, and this region which is right up there, what do you call this as? This is known as the radicle. Okay, this is going to form the root. And since the root is going to go inside the soil, it is going to be a rough journey for it, which is why it wears a cap and says that, Macha, please protect me. So over here, you have something else which is known as the root cap. So radicle is there, root cap is there, you have the plumule at the top, this is the hypocotyl and this is the epicotyl, and you have 2 cotyledons. This is the only thing that you have to remember about dicot. And now the picture that is given in NCERT is kind of different. So I'll tell you how to make that as well. So in the NCERT, it's given like this. You make the embryonal axis, okay? You make a drooping cotyledon like this, similarly another one like this, and tada, it's been done. So these are the two cotyledons. This is a hypocotyl having radicle and the root cap, and this is your epicotyl having the, what do you say, plumule out there. That's all. That's the only difference between this picture and this picture. This is for your understanding. So I'm telling you, mostly all the dicots which are there, mostly, I'm not saying every time, mostly all the dicots which are there, they utilize the endosperm completely to form cotyledons. In that case, they form dicots. So when you talk about a seed now, okay, this embryo is within the seed. What type of seed it is? It is known as non-albuminous seed. I'm trying to explain you something else also over here. This is known as non-albuminous seed. What do you mean by non-albuminous seed? Where the endosperm is not left back. Where the endosperm is not there, you will call this as non-albuminous seed. Always remember this. This is very, very, very important. Cool? Yeah? Exception is there, ma'am, of dicot that is non-albuminous, sorry, of a dicot that is having endosperm left behind. I'll talk about that. Listen to me. Before that, the dicots which are non-albuminous, that means does not have endosperm left back. What are the examples for that? Pea, groundnut, beans, all of it which you can break into two. Basically, they are dicots, that is why you can break into two. All of them, they are basically dicots. They have a dicot embryo and they are within a non-albuminous seed, where usually within the seed you do not have endosperm left back because the embryo has completely used it up for forming cotyledons. Exception of a dicot which is not going to be non-albuminous. It's not going to be non-albuminous. I'll write it here. Exception of a dicot, and that is what? Castor. Castor is a dicot which is albuminous. Albuminous means what? It will be having some endosperm left back. It is not going to utilize all of it for forming cotyledons. It will be having some endosperm left back. Please remember it. This is an exception, that is castor. It comes under albuminous seed, not non-albuminous seeds. Okay, so this is an embryo. Embryo is within the seed. I'm talking about seed. The seed is going to be non-albuminous when the endosperm is being utilized to form the cotyledons. Example is pea, groundnut and beans. Castor is also dicot, but it's not an example of non-albuminous seed. The reason being that their endosperm is still left behind. It becomes albuminous, not non-albuminous. I hope it's a little bit messed up, but I hope it's clear. I'll be talking more about it, don't worry. So this is the picture. This is the picture that is exactly given over here. Embryonal axis with two cotyledons is there in the case of dicot. Epicotyl above the cotyledon. Yes, and plumule is the stem tip. Hypocotyl below the cotyledons, and basically at the tip you have radicle or the root top. Root tip covered by the root cap. I told you about that already. Yes, and here is the root cap that is made. Now when you talk about monocot, things are going to be kind of different. I told you monocots, dicots, if they are known as non-albuminous, monocots which are there, they are going to be known as albuminous. Why? Because they will be having some endosperm left behind. So ma'am, you are saying that they will not form the cotyledons, they are not like them, those children? No, they will also form the cotyledon, but only one. That is why you call them monocot. So listen to me very, very carefully. Over here, when you talk about monocot, this is the embryonal axis, and they just have one cotyledon. And since they have only one, when we have only one thing out there, we probably take care of it very nicely, put their name and Chunnu, Punnu, Tunnu and all. Similarly over here, we have given it a name that is known as scutellum. It is going to be known as what? Scutellum. This is very, very, very important. Okay, it is known as scutellum, and basically this is the only one cotyledon which is there. Now, does it have endosperm left behind? Obviously, yes. It will also have some endosperm that is left behind as well. That is also equally there. Got it? Yes, ma'am. That's why do you call it albuminous seed? Yes, I call monocots as albuminous. Why? Because they have endosperm and also some amount of cotyledon. I'll call this albuminous. Why? Because they have endosperm. That's the reason, not because they have cotyledon. It's just for your understanding, I told you. Cool. Example of albuminous seed or basically monocots, it's basically you have barley, you have rice, you have wheat, all of them. And we saw an exception there. Castor. Castor is an exception. It's actually a dicot. but it comes over here. It has albuminous seed. That means it will be having endosperm left behind. It will be having endosperm left behind. So I'm talking about two different things, I know, but it's very important for you to associate it like this, otherwise you will not understand. Okay, so castor has two cotyledons, but it has still endosperm left behind. Over here, when I talk about others, they are usually monocots, have cotyledon only one, and endosperm also. Now here there are much more differences that is there. This is your embryonal axis. In the embryonal axis, Baba, over here at the top, what do you have? You have the plumule. Here what do you have? You have the radicle. Covering the radicle, what do you have? You have the root cap. Covering the root cap, what do you have? Something else. So in the case of monocots, you have everything extra. Since anyway they have saved some energy in not forming another cotyledon, they are going to have some extra things out there. So the first thing that they have, which covers their plumule or the shoot apex, okay, plumule or the shoot apex, which is going to be covered by something that is known as coleoptile. So always remember P over here and P above plumule, coleoptile. On the other hand, when you see over here radicle, right? Radicle of the root cap is covered by something else which is known as coleorhiza. It is covered by coleorhiza. So R over here, R over here. So coleoptile there, coleorhiza over here. They have extra things also. They have a scutellum as well. Please do remember this. This is also super important. Cool? Yes. Now over here, if I draw the picture, now in your NCERT, if you see the picture, you will be like, oh my god, this is not the picture that you drew. So I will show you how you make that. But before that, let's read it. So monocot embryo parts is one cotyledon, that is scutellum, lateral to the embryonal axis. Lateral, I mean parallel to the embryonal axis, you have what? The scutellum. Radicle and the root cap is enclosed by coleorhiza. Yes ma'am, done. And epicotyl with shoot apex and leaf promodia is inside the coleoptile. The top portion which is there is basically covered by the coleoptile. Done. Now we make this particular image, and this is very important for you to know. The reason being that you get questions to draw the monocot embryo. Yes, and by the way, monocot, example of monocot is also grass. Grass is also a monocot. Please remember, grass is also a monocot. Okay, so over here I'm gonna make it. So let's go. First of all, you have to make whale's mouth. Then whale has a fin. Then whale has a tail. And then whale has a smaller fin. Okay. Over here, you have to make a triangle like this. Come on. Yes. And shade it up. This is going to be your coleoptile. Then you make a smaller triangle and shade it up. This is going to be a shoot apex, basically the things that is going to form the shoot, or I can simply say the plumule. Then you go here, connect here and come back. What is this? By the way, this is the scutellum. You know, parallel to the embryonal axis, you have the scutellum, that is the cotyledon. So this is a scutellum. And this goes to only one tail and comes back. This portion which is there, it's going to be a radicle. Then you have the root cap. This last portion that you have is going to be your coleorhiza. And what is this? So actually they also tried to form cotyledon, but they were not successful. It got blasted away. So I'll say this portion which is there is known as epiblast. Sorry, epiblast. I should write it properly. Just a second. Epiblast. This is the picture that is given, and this is something that you can draw, and you will get your marks. Do not be worried. If you draw this, you are going to get your marks. The labelings and this part is important. You have to make sure you know that. Cool? Sorted. Draw a label diagram of LS of an embryo of a grass. What is grass? Grass is a monocot. So basically I've been asked about diagram of a monocot. This is a 3-marker question that have come in model question paper as well. Now we spoke about embryo, we spoke about the endosperm, and now we are going to go ahead with the last part of the chapter, that is what? The seed and its structure, and also the fruit and its types. So when you talk about seed, I told you the ovule is now transforming into a seed. Inside the ovule, to have your picturization, what do you have? You have the embryo, you have the cotyledon, okay, and this ovule has now transformed into seed, which is why the integuments which were there, they are also going to get transformed into what? Into the seed coat. The outer integument which is there, it is known as testa, and the inner one is going to be known as the tegmen. So tegmen, you just remember like that, it is inside something. So you have testa and the tegmen. So what is the seed structure? Seed coat is there, testa and tegmen. This is nothing but the ovules in teguments that you had earlier. Apart from it, inside the seed, obviously you will have the embryo and also the cotyledons are there. So cotyledon is there for food storage and it is pollen and legumes, and basically there is embryonal axis which forms the embryo later on. Right. The micropyle which is there, here I told you, if you remember, before fertilization it had a different function to form what? To basically help in a way to provide the entry of pollen tube. But here what is the use of it? Here, what is the use of it? Any idea? It forms the micropyle. It forms a micropyle. What is the use of having the micropyle? Basically, it helps in transfer of oxygen as well as water. It helps in transfer of oxygen as well as water. This is super important. Why you need oxygen water? The seed need oxygen water because the embryo is there within it and it needs to grow. For germination of the embryo to form a whole plant, you need water, because in the presence of water some hormones are going to get active and that helps in seed germination. You get it? So oxygen and water transport happens through the micropyle. Super important. Okay. Now types of seed we already studied about it. Non-albuminous and albuminous. Non-albuminous means endosperm, non-albuminous. Nahi hai, no, it's not there. Endosperm is completely used up to formation of what? Your cotyledons. Example is pea, groundnut and beans and everything. Albuminous endosperm partially remains out there. It is persistent. It remains out there. Okay. Example is wheat, maize, barley. All of them are actually monocots. Exception is only one, castor, which is a dicot. Okay. Now there's a term that is known as perisperm given over here, which is equally very important. So Baba, if at all you remember, we studied about this thing, right? Just give me a second, I'll talk about it. Where did we go? I think, yeah. We studied about having nucellus, and we told that nucellus is there for the nourishment of MMC and everything. This nucellus sometimes persists there. Says that I was there nourishing the MMC. I also want to see the embryo that it forms. So it's there, still there for nourishment of the embryo. When it persists like that, you have a term to it. You call it by a term, and that term is known as perisperm. It is known as what? Perisperm. So where are we? Let me come back. Yeah, we are over here. So this perisperm is what? Persistent nucellus, but it is not there in every other, in every other ovule that you see, only in some. So this persistent nucellus which is there, what do you call this as? Perisperm. And this perisperm which is there is found only in two plants according to your NCERT, according to its example, and I remember it as B square. B square means two of them starts with B only. Beet and black pepper. Beet and black pepper. These are the two things which you have, and this forms the perisperm, or basically this has the persistent nucellus out there which is known as perisperm. Later in this chapter itself, okay, just after 5 minutes, you are going to hear about pericarp. That is going to be different. So I think I should be talking about that here itself. What's stopping me from doing so? So this is about the ovule. Inside the ovule there is perisperm, right? That is persistent nucellus found in black pepper and beet. Now, when you're talking about ovary is outside, it forms the fruit. The wall of the ovary has a name, it is known as pericarp. This is pericarp and this is perisperm. Pericarp and perisperm. So sperm is inside the ovule, you can remember that perisperm. This is pericarp, it's basically ovary. The wall of the ovary together, it is known as pericarp. And if at all you want to distinguish into different parts, listen to me carefully, you just have to make a mango. When you make a mango, the portion that you peel off, that is known as exocarp. The portion that you gladly eat up, the fleshy portion, which is there. Okay, the fleshy portion which is there, just give me a second. Come on. Yeah, this is going to be known as mesocarp. This is going to be known as mesocarp. And finally, you have a hard portion out there. very hard portion. What do you call this as? This is known as endocarp. So what do you have? You have exocarp, that is the outer portion that you peel off, mesocarp that you eat, and endocarp, that is the hard portion. Inside this hard portion, you have what? You have the seed which is there. Seed will have its own seed coat and everything. That's a separate thing, but yes, that is the whole logic. That is the whole story about. Just give me a second. I think some issue happened. Yes. Now all of them, these three things together forms the pericarp, that is the wall of the ovary. Together we call this as pericarp. In some of the plants it could be fleshy like this, in some it is not. For example, peanut, do you find any fleshy part? There is no fleshy part. There's only a peel that you peel off. So that is the only wall of the ovary that we have. Nothing else. Rest everything is basically a part of the embryo. Right? So please remember that there can be fleshy or there can be not fleshy. So this is pericarp and this is perisperm. Both of them are different. This is ovary and this is for ovule. Okay. Now one more thing was given. When the seeds mature, its water content is reduced and seed becomes relatively dry. So when they mature, obviously, you know, if water and everything is there, there can be a chance of delay and everything. So until and unless they get a favorable condition, say they remain like that, they lose all their water, they remain relatively dry, and they will have only 10 to 15% moisture by mass, 10 to 15% moisture by mass. And this is, as I said, to make sure there is no decay, nothing happening out there. And whenever they get the favorable conditions and all, they will probably grow up like normal thing. Here in this particular case, the metabolic activity is also going to be very, very slow. When the seed matures, the metabolic activity is going to be very, very slow, as I said, until and unless it plans to germinate, if it gets the favorable conditions. Okay? As you can see, my energy is dropping down, but I'll keep going on. Why? Because you guys are listening to me. So distinguish between albuminous and non-albuminous seeds. Over here you have albuminous, non-albuminous. They retain some endosperm even after seed maturation. Albuminous, right? They have some endosperm. They do not have it. Mention that. Here, who provides nutrition? It can be endosperm and also one cotyledon. Here it's usually just cotyledon in non-albuminous. Example for both of them, and the thing is sorted. This is a question bank question, 2-marker question. It came. Please remember it. I've taught you with all my heart. Okay? Ayo, just a second. Yeah. Now, advantages of seed. So when we talk about seed, we studied about structure of it. We're talking about why is it good to have a seed. So listen to me very carefully. It provides independence from water. You know, seed is something that is a feature of big plants, as in basically the developed plants, gymnosperms and angiosperms. When you saw in algae, bryophytes and pteridophytes, you saw they need water for pollination and everything. But when it comes to seed, they do not need water for pollination. Pollen grain just has to come somehow. Whether it be wind, whether it be biotic agent, it can be anything out there. They necessarily do not say, I need water for pollination. So this is independence from water, and this is a good thing because not everywhere on earth you have water. It allows it to sustain anywhere. So first is independence of water. Pollination and fertilization do not rely only on water. That is the first thing. Second thing, dispersal and colonization. You know, seeds are very small. They can be very light. If an organism is coming up and probably by any chance in its fur, some seed is getting stuck, it can easily get transported to some other place as well. And by that, you get new plants everywhere else. So dispersal and colonization. Colonization means at any new point, it can form its own colony. It's very easy process. Independence from water, dispersal and colonization. The next one is food reserve. A seed makes sure, when you have a seed, it makes sure that embryo gets enough food. Protection is there, definitely. That is the next point, because there's a seed coat. Apart from that, a seed makes sure that inside it there is a cotyledon or probably some endosperm left behind, such that the embryo which is there, which is going to form the new plant, is having enough food. And finally, genetic variation. Seed is a product of what? Sexual reproduction. So obviously there can be a chance that there will be higher genetic variation. So these are the 5 points. This came not as a 5-marker question. Basically, in a 5-marker question, there was 3 plus 2 categorization, and in a 2-marker portion, it came. What is the advantage of seed? These are the advantages of seed. Independence from water, dispersal and colonization, food reserve, protection, and finally, the genetic variation. Okay, now, in your NCERT, 2 examples are given of seed which remain viable for very long. What do you mean by seed viability now? Seed viability means that they have the ability still to probably germinate and give rise to a whole plant. Okay, so some of them remain viable for 10,000 years. and some of them remain viable for 2,000 years. Still, it's a big number. The one that stayed viable for 10,000 years is right here, over here. This is found in the Arctic tundra region. This is known as Lupinus arcticus. It is known as Lupinus arcticus. It remained viable for 10,000 years. Okay, and over here you have the next guy, and I'm pretty sure you have seen it. Is the date palm plant. From here where you get the date. This is known as Phoenix dactylifera. This is known as Phoenix dactylifera. You have the scientific terms, you have to remember it. So this remained viable for 2,000 years. And this remained viable for 10,000 years. 10,000 and 2,000. Lupinus arcticus 10,000 years, it's found in the Arctic tundra, and Phoenix dactylifera date palm 2,000 years old, and it was found during Dead Sea excavation. Very, very, very important. Cool. I hope this is actually sorted. So, yes. And by the way, Phoenix dactylifera, which was there, it was found during the Dead Sea excavation during King Herod's time. During King Herod's time, that is the extra thing. King Herod, D, and date palm, Phoenix dactylifera, you can remember like that, 2,000 years old. King Herod cannot be too old, right? Cannot be 10,000 years old. So you can remember like that. Oh my God, I'm super tired. Okay, so one of the 2,000-year-old viable seed discovered during the archaeological excavation at King Herod's near the Dead Sea is what? Maize, sunflower, lupin, or Phoenix dactylifera. You know the answer. It is going to be Phoenix dactylifera. This is 2022 question. Yes. By the way, I'm reminding you once more. If you're someone who is probably taking Pariksha Vira, you will be able to solve all the questions related to KCET. I mean, I have solved more than this. All the questions actually which have come in KCET there. And there will be more interactive live sessions out there. So if you're really interested, make sure you check it out. Yes, yes. Now we come to the fruit part of it. In fruit, we have discussed one part of the story, that is the wall layers of the fruit. We discussed it already. It is known as pericarp, and it has exocarp, mesocarp, and the endocarp. Now, there are two types of fruits. Okay, I told about fleshy and the non-fleshy ones, right? Fleshy, where you have the mesoderm, thick mesoderm and all, it's mango, guava and all. Dry, such as mustard and groundnut. They do not have a separate layer that you can see. Oh, this is exocarp, this is mesocarp. You cannot see that. Okay, now we'll come to the fruits. There are three different types of fruits that you need to remember. There is true fruit, where basically this is the ovary, this is the ovule. Ovule becomes the seed, ovary becomes the fruit, and this is known as true fruit. Nothing else is participating in the formation of a fruit. Only the ovary, only the ovary. Okay. Next is false fruit. False fruit means basically over here, suppose I have the thalamus onto which you have the beautiful flower which is resting. Just give me a second. Okay, just give me a second. Yeah, and yeah. You are expecting the ovary to form the fruit here, right? But here, some other parts are also taking participation in the formation of fruit. I'm not saying ovary is not taking part. Ovary is definitely taking part, but apart from it, some other parts of the plant is also taking participation in the formation of fruit, and this is known as false fruit. Now, it can be thalamus like this, or it can be pedicel of the plant. It can be anything along with ovary that helps in the formation of fruit. This is known as false fruit. Where do you see it? You see it in apple, you see it in strawberry, okay, and finally you see it in one more thing, that is cashew. Apple, strawberry and cashew, ASC. Apple, strawberry and cashew. This is all where you can find false fruit. Why, ma'am? How can you clearly say that? So I'll show you a picture, and I think all the things would be more clearer to you. So if you see the apple, the thing that you actually eat, the fleshy part, is the thalamus, because that transformed into the fruit. What about the ovary? Ovary is also there. Inside that, you might have seen towards the seed, there's a thick region out there. That is basically your ovary that is participating in the formation of fruit. Inside that, you would be having your seed. So there is definitely mesocarp, there is endocarp also, there is exocarp also. Not defined clearly, exocarp would not be there defined clearly. But yes, you have mesocarp, endocarp, both of them are basically parts of your ovary. Then you have thalamus, that's the actual part you eat, which is why I told you that here fruit formation is happening with the help of other parts of the plant. Cool? Cool. Now, the next thing which is there. So false fruit is done, true fruits is done. Just give me a second. Okay. Now, example is important: apple, strawberry, and cashew. In some of them, this pedicel forms takes help in the formation of a fruit. In some, it's basically the thalamus, apart from the ovary. Okay. So here, I just want to clear up. Through false fruit, thalamus contributes to the formation of fruit. No, it's not like that. Other floral parts apart from the ovary also, right, also participates in the formation of fruit. It's not that other floral parts makes the fruit. Other floral parts apart from the ovary also participate in the formation of fruit. That is known as false fruit. Example is apple, cashew, and strawberry. Now we come to the last thing, that is known as parthenocarpic fruit. For the parthenocarpic fruit, things are going to be quite different. We were expecting that, okay, this is ovary. When ovule undergoes fertilization, that's when it becomes seed and this becomes a fruit. But here, please understand, I'm writing it clearly and you need to know this. Without fertilization, ovary is transforming into a fruit. How? How can that be possible? Because of some hormones. So mostly these hormones are auxins and all. Okay, they are auxins that takes participation in this, but yes, it's basically hormonal. And in this case, you only think, if there is no fertilization, do you think seed will be formed? Ovule will not transform into seed because you need fertilization for that. So over here, you will not see seeds. So they are going to be seedless in nature, and as I told you, it's hormonal. It's hormonal process out there. Example, banana. Example, you have banana. Ma'am, can we induce parthenocarpy? Yes. Matlab, can you cause parthenocarpy? If at all a plant is not parthenocarpic, can you make it parthenocarpic? Yes. By inducing hormones. By giving some hormones, you can make it as parthenocarpic. Got it? This is also equally important. So over here, parthenocarpic fruits develop without fertilization. Example, banana can be induced using growth hormones. They are seedless fruits. Please remember that. Parthenocarpy, fruits without fertilization. Now we are going to talk about apomixis. That is going to be different. Parthenocarpy is about fruit, ovary, okay, fruit without fertilization, and over here you will not find the seed. Now the last part of the chapter, now I'm super excited. Come on. By the way, giving reasons, this is a 2-marker question that came. Micropyle remains as a small pore in the seed coat of the seed. Why? For transfer of oxygen as well as water for germination. Apple and cashew are not true fruits because they are formed from the other floral parts of the plant apart from the ovary. Okay, apart from the ovary. Okay, so write it like that. True fruits develop only from the ovary after fertilization. In apple and cashew, the edible part develops from the floral parts other than the ovary. Hence, they are called as false fruits or accessory fruits. Cool? Yes. And now, differentiate between pericarp and perisperm. Pericarp is what? The outer layer of the ovary. You differentiate into 3 parts: exocarp, mesocarp, endocarp. Exocarp is the outer layer, skin of the fruit. Mesocarp is the middle layer, fleshy in many fruits. Endocarp is the inner layer, hard or membranous. Right? Perisperm is what? Persistent nucleus is known as perisperm, found in black pepper and wheat. If you want to give example with the help of diagram, make a diagram of the mango. Now, in the given diagram, identify the parts labeled as A, B, C and D. Now, this is a picture of actually a monocot seed carrying the monocot embryo. Okay, this is a monocot seed carrying the monocot embryo. So, if I say this is the embryonal axis, where is the scutellum? Scutellum will be just parallel to this. So, B is going to be the scutellum. This is the embryonal axis. Here you can have coleoptile or plumule. Here you will be having coleorhiza, radicle and everything. Okay, whatever is left back over here, this is the endosperm. This is what? Endosperm. I told you here in monocots, endosperm persist, right? So we'll go one by one. We do not know what C right now. So over here, I told you B is going to be this. But before, if I do not know about all of it, I know this is an embryos, monocot embryos, embryonal axis. Then A can be coleoptile or it can be plumule, right? Coleoptile or plumule. You just have 2 options with coleoptile, you keep them. You just have two options with coleoptile, you keep them and you remove the other options. Just a second. You remove the other options. Done, ma'am? Done. Okay. Now D1. D1 is going to be either coleorhiza or radical or root cap. D1, coleorhiza, radical or root cap. You just have two options, pericarp, it cannot be a pericarp. So obviously it has to be this one. A is going to be your answer. What is B over here? I told you it's the scutellum. What is C over here? The last one. It's going to be the pericarp. So basically you have got here the embryo, the ovule basically, this ovule having the embryo, and it's covered by the seed coat. Seed coat is not given as an option over here. So the next covering which we have is what? It is going to be the pericarp. That is the covering of the ovary. Yes. And finally, you have apomixis as well as polyembryony. Let's get it, guys. So those of you probably reached until this point, my God, I should actually give you a salute. So listen to me carefully. Apomixis is a whole different scenario, something that we have not seen until now. Parthenocarpy is what? Formation of fruit without fertilization, right? There you did not have seed. Apomixis is going to be different. Apomixis, you have the ovary, you have the ovule, and I think it's one of the most toughest topic for you guys, so I'll try to make it easy. This is the ovule, right? Inside the ovule, you have the embryo sac. So Baba, until now what we have studied, that synergids, all of them, egg cell, all of them are going to be haploid. But just think of a scenario where for some reason, in the plants, the reductional division did not take place. That means meiosis did not take place. Do you think the number of chromosomes will become less over here in the egg cell? No. It remains diploid. It does not become half, rather it remains as diploid. Now if it is already diploid, do you think you need a male gamete to form a zygote? Because usually female gamete is haploid and it waits for the male gamete such that it can become diploid to form a zygote. Here you do not need that. Why? Because I am already diploid. Why can't I just form the zygote by myself? Can I do that? Yes, but only in some plants it happens. In grasses and also in plants that comes under Asteraceae. This happens where, my dear children, the egg cell is already diploid because it has been formed without meiosis for some reason. Now if it is already diploid, it is going to form what? Zygote. Now if it forms a zygote, can I now call the ovule as a seed? Can I call the ovule as a seed? Yes, ma'am. But fertilization did not take place. I am not saying fertilization took place. Here also no fertilization took place. There we studied no fertilization took place and there you got the fruit. But here you are getting a seed without fertilization. Why? Because you already got a zygote without fertilization, because there was no meiosis happening up there. This is known as apomixis. I'll repeat it. Usually you have meiosis, you have haploid number of chromosomes over here, you need the male gamete such that haploid comes together, it becomes diploid zygote. Only when the zygote is being formed, only when this particular zygote is being formed, that's when I call the ovule as a seed. Until then I cannot call it. But here it's happening without all this thing. Why? Because meiosis did not take place. It's already diploid in nature. This diploid thing transforms into zygote. And that's all, without fertilization. No fertilization. This ovule is now known as what? A seed. This is apomixis, formation of ovule, formation of seed without fertilization. Now it can be done in this way, formation of a seed without fertilization. The only target is that I need to form zygote, I need to form embryo. That's the only target. If I've done it without fertilization or with fertilization. Without fertilization happens, it is known as apomixis. This is one scenario. This is apomixis, and there is scenario 2 also. Apomixis can also happen like this. If you remember, you have the ovary, you have the ovule. Inside the ovule, you have the cells of the nucellus. Sometimes it persists, I told you. Yes, then you have the embryo sac. Inside the embryo sac, everything is normal only. Everything is normal. That is basically you have the egg cell is there. Okay, sorry, you have the synergids and then the egg cell. The egg cell is going to be haploid. The egg cell over here is going to be haploid. A male gamete comes, makes fusion. Okay, it forms the zygote and everything. That is there. But apart from it, listen to me very carefully. Over here, these cells, the cells of the nucleus, can also get inside and form the embryo. That is happening without fertilization. Without fertilization, basically they come inside, they are nucleus, basically the cells of the nucleus, they are already diploid in nature. When they come inside, they are going to form a diploid thing only, no? So they are forming diploid embryo. Embryo means multiple cells are there. You cannot say zygote because they are diploid, they are coming inside and they are dividing and coming inside. They directly form embryo. Again, we are forming embryo without fertilization. So can I say this is also a way of apomixis? Yes. In this way also, apomixis can happen. It can happen like this without reductional division. It can happen like this where the cells of the nucleus can come inside and make embryo. Well, if it happens like this, let me tell you, this is also an example for another term that is known as, polyembryony. This is also known as polyembryony. Polyembryony means multiple embryos being formed. Do not get confused. I am telling you, apomixis can happen like this. Apomixis can happen like this. If it happens like this and multiple embryos are being formed, this condition is known as polyembryony. I am not saying every time apomixis happens, it is going to form polyembryony. No, this is just an example. Okay, here it happens. That is why I am telling you that polyembryony is taking place over here. Now, where do you usually see polyembryony, multiple embryos being formed? This is a case of citrus fruits. Okay, in citrus fruits such as mango or probably lemon and things like that, you will be finding polyembryony. Okay, and apomixis, broad example, is basically grasses and Asteraceae. Grasses and plants belonging to Asteraceae family. So apomixis can happen like this and this. The bottom line is you have to form zygote or embryo without fertilization. Are you doing it? Yes. So then that's cool. That is apomixis. It can happen like this or like this. If it happens like this, you get multiple embryos also. This is known as polyembryony. Example is citrus fruits. Okay. In general, it actually happens like this. Now over here, do you see any fission of gametes? No. So this is sexual reproduction? No. It mimics sexual reproduction. It is mimicking sexual reproduction by forming the zygote. But it is actually asexual reproduction. Remember, it mimics sexual reproduction by forming what? Zygote. But it is actually what? Asexual reproduction. It is actually what? Asexual reproduction. This is very, very, very important. Got it? Sorry, my legs were pairing. Got it? So mimics sexual reproduction, but it's actually asexual reproduction, which is why, remember one more thing, all the organisms which are going to be formed will be like clones. Will be exactly similar. Will be what? Clones of the parent plant. They are going to be similar, ditto, same. Works? I hope that's done. Yes. Okay. So that's all about it. So yeah, and now I'll read it out, okay? Apomixis is seed formation without fertilization. Example, Asteraceae grasses mimic sexual reproduction, but is actually asexual reproduction. It can happen in two ways. Diploid egg cell forms without reduction division. It develops into an embryo without fertilization also. Nucleus cells divide, enter the embryo sac and develop into embryos. This is found in citrus fruits and mango. And basically this is a situation where multiple embryos are being formed. This is known as polyembryony. Okay, genetic nature, apomictic embryo are going to be clones. So why is this going to be useful for the farmers as the last part of the question? And this is a question that actually came in the question bank also in second PUC. So why is it important, apomictic seeds? Listen to me. If I'm a farmer, basically you are a farmer, and you want plants with good quality. So what do? Hybridization is being done and you get hybrid seeds. Usually you buy hybrid seeds from outside. These hybrid seeds are very, very costly. And suppose I have hybrid seeds. I put it, okay? I put it over here and the plants grew. Just give me a second. And the plants basically grew out there. what is going to happen when they are going to grow? After this, they are going to undergo sexual reproduction because it's a natural process. Sexual reproduction. Will they have their hybrid characters now? After the end of sexual reproduction, I cannot guarantee that, because two things are coming together. They might lose it also. This is nothing that I can promise from my end. So hybrid vigor cannot be maintained over here. Hybrid vigor means the hybrid nature may not be maintained over here. But if I have an apomictic seed, does it grew up? Does it rely on some male plant to come and fertilize it? No. It is apomictic. It does not need sexual reproduction. It is going to grow into a new progeny, new seed, and that seed will be exactly like the parent plant. So in that case, the hybrid nature which was there in this particular plant, if this plant is really good, that vigor is going to be maintained throughout. So that is the first thing. Hybrid vigor is maintained. Farmers can reuse apomictic seeds every season without the loss of yield or quality or segregation of any character that is going to occur. Next is cost effective. Farmers do not need to buy hybrid seeds every year because you have apomictic seeds and they are going to go. Next thing is uniformity in crops. If I say that this is the crop I wanted, you'll get everything the same way because they are going to produce clones. So these are a few points which are super important for this, and I hope I was able to explain you this. And with this, we complete our one shot with, I think, 1 hour, sorry, not 1 hour, 3 hour 44 minutes of lecture. But I hope each and every thing that I said makes sense to you and it was helpful. Well, if it was, definitely let me know in the comment section. And my dear children, I just want to wish you the best for everything that comes up. Do not be demotivated. You can do it. And we will be coming up with more one-shots, more videos, and everything that is possible to make you guys be happy. And I hope I justified the last point. I hope I justified the last point over here, that you are tired definitely for sure, but you will be the happiest because we have covered one chapter, that is sexual reproduction, flowering plants, 13 marks guaranteed, 3 to 4 marks guaranteed. Yes, and FA1 full marks. So that's all. Bye-bye. Lots of love from Jasmine Mam. Take care.